tackling with new sources. In fact, in the Philippines, we just had our 7,100 cases as of yesterday. Okay, so this pandemic has indeed brought unprecedented challenges to our public health and has caused economic and social disruption. But on the lighter note, this pandemic has also triggered great opportunities to pursue new scientific knowledge and research in various fields of microbiology and biotechnology. In this Cluster 2 Symposium, some of these innovations in the new normal will be tackled by our speakers. I am sure that today's symposium will be filled with interesting discussions on the role of microbiology and biotechnology in providing possible solutions to address global challenges. I would like to commend the Cluster 2 Committee, headed by Dr. Noel G. Sabino, Dr. Lucille C. Villegas, Professor Christian Jordan de la Rosa, and Mr. Robbie Vasquez for successfully organizing this online symposium. I would also like to take this opportunity to invite you all to the 8th Asia Pacific Biotechnology Congress and the 50th Annual Convention and Scientific Meeting of PSM this July 21 to 24, 2021. The theme of this year's virtual convention is Microbiology and Biotechnology, Scientific Opportunities to Address Global Challenges. Distinguished experts from Japan, Taiwan, Pakistan, USA, Spain, and the Philippines will present new innovations in microbiology and biotechnology amidst the global challenges brought about by this pandemic. Lastly, I would like to extend my gratitude to the Institute of Biological Science, College of Arts and Sciences, University of the Philippines, Los Baños, for co-sponsoring this activity with us. A special thanks to our partners, the Procter & Gamble Philippines, ESCO Philippines, 3M Philippines, and Defensil Antibacteria for making today's activities possible. Mabuhay and stay safe, everyone.
Thank you, Professor Joel Carnesta, PSM President. At this point, we will jumpstart our speakers, right? So to introduce our first speaker for this morning, I would like to call on Dr. Lucille C. Villegas. Ma'am Lucille?
Yeah, good morning, everyone. I am very pleased to introduce to you our first speaker for this PSM Cluster 2 Symposium, a colleague, a collaborator, a neighbor, and a very good friend. Our first speaker is an associate professor of the Microbiology Division, Institute of Biological Sciences, College of Arts and Sciences, University of the Philippines, Los Baños. She obtained her Bachelor of Science degree in biology, major in microbiology, cum laude, in 1998, and her Master of Science degree in microbiology in 2007, both from UPLB. In 2011, she was the third prize winner in the National Academy of Science and Technology Talent Search for Young Scientists for her collaborative research work on rice tungroviruses. She has been in the teaching profession for 20 years and is a 5-time recipient of the IBS Outstanding Teacher Award based on student evaluation for teachers. She has handled graduate and undergraduate courses in microbiology, including general microbiology, advances in microbiology, microbial ecology, microbial genetics, and virology. Her current research works include the characterization of tungrovirus resistance in rice, virus drought stress interaction, dynamics of rice tungrovirus interaction, and the use of bacteriophages as biocontrol agents. For this morning, she will be sharing with us her work on bacteriophages as promising biocontrol agents against soft rot Pectobacteriaceae of high-value crops. Ladies and gentlemen, let us all welcome our first speaker, Professor Jamie R. Encabo.
Thank you very much, Dr. Villegas, for the introduction, and thank you very much to PSM for this opportunity to share with you this morning our work on bacteriophage research. Viruses of bacteria, more commonly known as bacteriophages or simply phages, have been proposed as an alternative, environment-friendly plant pathogen control agents in a process known as phage therapy. Now, phage therapy involves the application of specific phages to specific ecosystems to reduce the population size of specific bacteria. Now, over the past years, the promise of phage therapy as a means of combating plant disease has been explored with increasing enthusiasm. So in the next 30 minutes, allow me to share with you our learnings and interesting findings on soft rat pathogens and on the potential use of phages as biocontrol agents.
Good morning, everyone. In the next 30 minutes, I will be talking about bacteriophages as promising biocontrol agents against soft rot Pectobacteriaceae of high-value vegetable crops. 5-year data from the crop statistics of the Philippines show the volume of production for 5 high-value crops. Now, highest production volume was recorded for onion, followed by cabbage, potato, carrot, and lettuce. Now, vegetable production in the country has been characterized by a yearly fluctuation in the volume of production and a continuous decline in the area of land utilized for cultivation. Now, data on cabbage show that the Cordillera Administrative Region or CAR remained the leading producer of cabbage with 78.5% share to the country's total output in 2019. And this was followed by Central Visayas with 6.4% share and Northern Mindanao with 5.5% share. However, notice that production volume in the area harvested for cabbage decreased from 2014 to 2018, followed by an increase in 2019. No? So there are actually a number of factors that limit the production of vegetables, and this include climatic and soil conditions, land availability, per capita consumption of vegetables, and of course, we have pests and diseases. Now, soft rot is a destructive disease of vegetables and has always been a perennial problem in the Philippines. Soft rot has a wide host range, infecting vegetable species belonging to all families and causes a greater total loss of produce than any other bacterial disease. Now, the disease is considered as a major post-harvest problem in vegetable crops, accounting to about 40 to 50% loss, and is therefore a major concern to address in the Philippines. However, effective control measures for this disease is yet to be developed. Now, bacterial soft rot is a disease complex caused by a multiple genera of gram-negative and gram-positive bacteria, with Pectobacterium endicea being the most widely studied soft rot bacterial pathogens. Now, quantitative losses caused by soft rot Pectobacteria are greater than the losses caused by other bacterial plant pathogens. Now, the name of the disease came from the characteristic soft decay of fleshy tissue, which terminates into watery or slimy mass. And the most distinctive feature of soft rot pathogenesis is the coordinated production of a large number of plant cell wall degrading enzymes, which makes soft rot Pectobacteria very effective in decaying plant tissue. Now, the disease occurs worldwide and can be found on crops in the field, in transit, in storage, and during marketing, resulting in great economic losses. The taxonomic classification of soft rot Enterobacteria has been revised several times in the recent years. Now, soft rot bacteria was first characterized in the early 20th century and for decades were known as members of the genus Erwinia. Soft rot pathogens were later moved out of the genus Erwinia and the genus Pectobacterium was created, which includes the soft rot species. Later on, subspecies of Pectobacterium carotovorum were raised at species level, giving rise to, for example, you have Pectobacterium wasabiae and Pectobacterium atrosepticum. Furthermore, Pectobacterium chrysanthemi, previously recognized as Erwinia chrysanthemi, was separated from Pectobacterium and the new genus Dicea was formed. And finally, Pectobacterium arrhydiarum was described as a novel species consisting of soft rot pathogens, mainly infecting monocotyledonous plants. So that was reported in 2012. Now, SRPs are currently classified under the order Enterobacteriales, family Pectobacteriaceae, containing 5 genera, namely Brinaria, Dicea, Lonsdalea, Pectobacterium, and Sodalis. Now, this slide would show you a list of the 24 recognized species of the genus Pectobacterium, and you also have the 13 species recognized in the genus Dicea. No? So, of particular importance would be the soft rot species that we focused on in this particular work. So you have Pectobacterium arrhydiarum, P. atrosepticum, Pectobacterium brasiliense, Cacticida, Carotovora, and the rest, Carotovorum and the rest. No? So in the Philippines, there's limited information on soft rot disease. In fact, the only comprehensive study on bacterial soft rot was done by Daeng Subha and Kimyo in 1980. So in their work, they were able to isolate 80 soft rot associated bacteria, and they have identified Erwinia carotovora as the predominant species during that time. No, although there are other bacterial species that were identified in their work, and this includes Bacillus subtilis, Pseudomonas alicola, Pseudomonas cepacia, and Xanthomonas capsicei. And then host range of the bacterial isolates were examined in their study using 13 different vegetable crops. And what they observed was that their Erwinia isolates varied in their host range, and there was about 70% of the isolates having wide host range, infecting 7 to 11 types of vegetable crops. Now, control methods for soft rot disease include sanitation of packing house, lowering of storage temperature and humidity, host resistance, and other physical and chemical measures. Now, current disinfecting agents used include hypochlorites, chlorine dioxide, copper, quartz, and hydrogen peroxide. No? So the capacity of bacterial pathogens to develop resistance to chemicals and concerns about the negative impacts of chemicals on human health have promoted the exploration of environmentally safe bactericides against soft rot disease. So in the recent years, there were several published reports on the use of plant extracts, gamma irradiation, endophytic bacteria, and of course, bacteriophages against soft rot bacteria. And in the past 3 years, our work focused on phage-mediated biocontrol of soft rot disease. So in this particular work, we identified and examined the diversity of soft rot Pectobacteriaceae and their associated phages. We also determined the lytic activity of phages against selected soft rot Pectobacteriaceae and evaluated the efficacy of selected lytic phages against selected pathogenic soft rot Pectobacteriaceae. So in this work, we focused on 5 major crops. And this includes lettuce, carrot, cabbage, Chinese cabbage, potato, and onion. And in the past 3 years, we collected a total of 915 samples from 60 collection sites in the 34 municipalities of the 11 provinces we visited all over the country. So looking at this figure, this would show you that the majority of the samples that we collected came from Luzon, so that's around 60%, and then 36% were from Visayas, and 6% were collected from Mindanao. Now, for our research strategy, we used an identification pipeline for SRPs, and this involved the isolation and purification of bacterial isolates, followed by cultural and biochemical tests. And then we also did some infection assays using tissue discs of selected vegetable crops to determine the pathogenicity of the bacterial isolates in our collection. And then we did some molecular characterization using genus-specific markers, 16S sequencing, ERIC and rep-PCR, phylogenetic analysis, and also host range assay, no? So to determine particular crops that were infected by selected soft rot pectobacterial isolates. Now, alongside with this, we also used uh, a strategy for phage work that included the isolation and purification of bacteriophages using spot tests and plaque assays. And then this was followed by bacteriophage characterization, wherein we observed and described plaque morphology, determine the host range of bacteriophages, determine their optimal multiplicity of infection, as well as the burst size. No? So these particular parameters were important in selecting bacteriophages that are suitable for biocontrol applications. And then, of course, this was followed by experiments that would focus on the assessment of the lytic activity of selected phages. And this employed in vitro challenge tests and semi-implant assays. Now, for the bacterial culture collection, since there's really very limited information, particularly on about the predominant bacteria associated with vegetables in the Philippines. So for the 900 plus samples that we collected, no, so we used a combination of semi-selective and differential media to screen for soft rot pectobacteria. So initially, we grew the isolates in crystal violet pectate medium. No? So this is a very useful semi-selective media. So it allows the growth of gram-negative bacteria. And then using this medium allowed us to screen for pectinolytic bacteria based on cavity or pit formation. Now, the second medium that we utilized was the tree medium based on a publication by Cado and Hesketh in the 1970s. Now, this particular medium allows differentiation between SRPs and non-SRPs based on sugar utilization. So, this particular medium contains arabinose, and then soft rot pectobacteria such as Pectobacterium species would produce a distinct red coloration, which can be differentiated from non-SRPs, no, such as Xanthomonas, which will produce that distinct blue-colored colonies. Now, of the 1,268 bacterial isolates in our collection, we observed that there were around 913 that produce cavities or pits in CVP medium. And then of the 913 CVP positive isolates, we found that there were around 348 bacterial isolates that produce red colonies, no, in D3 medium, which we tagged as the putative SRPs. No? So after this initial screening, we were able to summarize the distribution of the bacterial isolates in our collection. So as you can see in this slide, no, so the isolates were categorized, no, so this the distribution is presented per crop, per location, no, per location where the isolates were collected. So this figure would show you actually that more than half of the isolates per crop at a particular location were found to be CVP positive. So that means there are a number of isolates that were identified as pectinolytic bacteria. But then only a small number of these CVP positive isolates were found to belong to the soft rat pectobacteria group. No? So these are denoted by the orange color in the bars that you would see in this figure. So in summary, what this figure would tell us is that the soft rat pectobacteria that we have isolated, no, particularly the ones present in our collection, would only comprise a minority, and that there are also other uh, soft rat bacteria, no, soft rat bacteria that are not included to the SRPs, no, that we were able to isolate from different soft rat disease environments. Okay. Now, what we did next was to examine the pathogenicity of the 348 D3 positive bacterial isolates, and then what we observed that there were around 163 isolates, around 47% of the D3 positive isolates were pathogenic to their original hosts. So this indicated that more or less around half of those D3 positive isolates produced the characteristic soft rat symptoms when they were back inoculated to their original host crops. No? So the figure here would show you the distribution of the pathogenic isolates, no, per crop following pathogenicity testing. And then what we did next was to identify the 163 pathogenic isolates using PCR-based molecular markers. So we did PCR using Pectobacterium decaya specific primers, and we were able to identify that all of the 163 pathogenic isolates belong to the Pectobacterium decaya group. No? So of the 163 SRPs that we identified, what we did next was to do a 16S rDNA sequencing to confirm their identity. No? So out of that 163 SRPs, we sequenced 101 of them for species identification, and then results of sequencing analysis showed that the most predominant Pectobacterium species in our collection was Pectobacterium aeroidearum. No? So out of that 101 sequence isolates, 86 were identified as Pectobacterium aeroidearum, and then we also identified Pectobacterium brasiliense, Pectobacterium carotovorum, Pectobacterium betabascularum, parmenteri, and a few isolates belonging to the genus Dicea. Now, what we did next was to analyze, no, the gene sequences of the selected Pectobacterium isolates from our collection, so that 98 Pectobacterium sequences and 3 Dicea sequences were compared, no, using 19 sequences of Pectobacterium Dicea as reference strains, and the phylogenetic analysis revealed that the Philippine SRPs are closely related, no? So majority of them are closely related, just like what you see in this figure, and they're mostly similar to P. aeroidearum sequences. No? Although there were 3 unique sequences of 3 bacterial isolates that were obtained from onion and cabbage. Now, in this figure, certain colors would also give you information about the host range of the isolates, no? And then it will tell you about the specific specificity of the bacterial isolate to a particular host, no? So red colors would tell you that the particular isolate would be pathogenic to that particular crop, while the yellow color would indicate non-pathogenicity of the isolate to the crop tested, no? So looking at the host range results, no, analysis revealed that some isolates actually can infect around 2 to 8 crops based on host range analysis. And looking at this information, we also observed that more than 50% of the tested isolates would infect 4 or 5 crops, which indicate that majority of the soft rot pectobacteria in our collection have wide host range, no? So we also examined the diversity of selected Pectobacterium isolates using rep-PCR fingerprinting strategy, no? So in this figure, we observed that both ERIC and rep fingerprinting revealed significant genetic variation among P. arrhydiarum isolates. So if you can look at this panel, ERIC fingerprints defined from selected P. arrhydiarum isolates. So we have here a number of P. arrhydiarum isolates which showed significant genetic variation in their banding patterns, no, based on ERIC fingerprints. And then looking at the dendrogram that we generated, no, so this would also show you the genetic relationships among the Pectobacterium isolates based on ERIC fingerprints. So it really tells us a lot about the significant genetic diversity of the pathogens that we have isolated. Now, as far as the bacteriophage collection is concerned, we were able to isolate a total of 138 phages, and then based on plaque morphology, we observed that the majority of the phages in our collection produce clear plaques, no? So 110 phages were found to produce clear plaques, while 28 of them produce turbid plaques. Now, of particular interest would be lytic phages, no, characterized by clear plaques, because these are the ones which would be suitable for biocontrol applications, no? So aside from plaque morphology, we also examined plaque diameter, and the phages in our collection also varied in terms of their plaque sizes, no? So a number of them produced plaques with a diameter of less than 1 millimeter, and then we also have phages producing plaques of with diameter of 1 to 1.9, and then we also have phages forming large plaques that would range from 2 to 3 millimeters, and those that would produce plaques greater than 3 millimeters. Now, in terms of phage source, 81% of the phages in our collection were actually obtained from symptomatic tissues. No? Although we also collected environmental samples in the form of sewage, soil, and vegetable washing. So we were also able to isolate a number of phages from these samples. Now, in terms of specificity, we observed that the majority of the phages in our collection were able to specifically infect Pectobacterium carotovorum. So there are about 69 phages out of the 138 phages in the collection that were found to infect P. carotovorum. Now, 35 of them were found to infect Pectobacterium brasiliense, and 24 were found to infect Pectobacterium arrhydiarum, and 10 infecting P. cacticida. Now, just to show you the variant morphologies of selected phages in our collection, a number of them were found to belong to the family Myoviridae. So these are the phages that would exhibit the typical head and tail structure, no, of myoviruses. And then we also have like one in another phage, one in another two phages that would belong to the Cyphoviridae and the Podoviridae family. No? So these are the ones also characterized by that head and tail structure, but compared to the members of the Myoviridae family, members of this family would have this characteristic short tail. Now, moving forward, we also tried to examine the diversity of the phages in our collection based on biological characteristics, no, specifically plaque morphology and host range. No? And then one very important observation was that morphologically similar phages infecting the same Pectobacterium species were found to group together. Now, interestingly, this analysis revealed to us four clusters of phages in our collection. So, of particular importance would be cluster 1 that would include super BHR phages. So, by super BHR phages, we mean broad host range phages. So, these are uh, of particular importance because broad host range phages are the ones capable of infecting several species of Pectobacterium. So these were found to group together in cluster 1, and then most of the phages in cluster 1 were isolated from Benguet. And then cluster 2 included phages infecting P. aeroidearum. No? So most of them are narrow host range phages infecting only P. aeroidearum, and then they were found or they were isolated rather from Benguet, while cluster 3 phages included phages that were found to infect Pectobacterium carotoverum, while cluster 4 included phages that were able to infect Pectobacterium brasiliense and Pectobacterium carotoverum. So this analysis indicated that the groupings of isolates correlate with their host range and geographic origin. But let me just mention again that this analysis only utilized certain biological properties of phages, particularly plaque morphology, which included size and transparency, as well as host range. Now, to determine the lytic activity of selected phages in our collection, no? So we focused our attention on specific phage isolates, no? So one particular set of phages that we zeroed in was those that were found to infect Pectobacterium carotoverum. So we have phage A3, T1, and J3. Now, these phages were isolated from tissues, soil, and sewage sample. We were able to characterize these phages. They varied in their optimal multiplicity of infection and their burst size. No? So the multiplicity of infection is actually the ratio of the number of bacteria relative to the number of phages, no, in a particular phage that you are characterizing. Now, what we observed was that this P. carotoverum phages were stable at a temperature range of 30 to 50 degrees centigrade, as well as a pH range of 4 to 9. Now, they vary in their latent period, so that would be somewhere around 10 to 30 minutes, no? And then in terms of their lytic activity, we observe that these are narrow host range phages able to individually lyse only P. carotoverum. No? So that means their lytic activity is specific against Pectobacterium carotoverum. No? So what we did next was to analyze or to evaluate the efficacy of this single phage suspensions against Pectobacterium carotoverum. No? So in vitro challenge tests, we used single infection treatments. No, so we mix bacteria with individual phages at particular multiplicity of infection, and then what we observed was that there was bacterial inactivation for all three single infection treatments as early as 1 hour post inoculation. Now, interestingly, in semi-implanta assays, we also observe that the protective effect no, brought about by this single infection treatments with these phages range from 30 to 60%, which indicated a significant reduction in tissue maceration caused by Pectobacterium carotorum. Now, this was followed also by an experiment which evaluated the efficacy of phage cocktails. So, in this work, we used a combination of 2 or 3 phages, and then we performed in vitro challenge test and in-planta assay. So, similarly, with this phage cocktails, we observed that bacterial inactivation was observed as early as 1 hour post inoculation no, at low multiplicity of infection, and more so, bacterial inactivation was also observed at high multiplicity of infection. And the results of semi-implanta assays also demonstrated that these lytic phages were really highly effective against Pectobacterium carotoverum at low and high multiplicity of infection, whether in 2 phage or 3 phage combinations. No? So these were very interesting results as far as cocktail evaluation is concerned. Now, another interesting observation was that we performed an experiment to determine the protective effect of this Picarotoverum phages. No? So in this particular work, using phage A3, D1, and J3, what we did was to vary the time of phage application relative to bacterial inoculation. So in a separate setup, we inoculated potato slices with the phages 30 hours and 3 hours before bacterial inoculation. And in several setups, we also inoculated potato slices at 3 hours and 30 hours post bacterial inoculation. So interesting observations were as follows. No? So what we observed was that all 3 phages were effective in preventing soft rot caused by P. carotoverum. So the tissue maceration was not observed for this particular treatments when phages were applied no, prior to bacterial inoculation. However, inhibition of Pectobacterium carotoverum was only observed for phages A3 and D1 from 0 to 3 hours phage application following bacterial inoculation. So it appears that at this particular MOI of 10, no, so we saw that the phages would seem to provide a protective effect no, against soft rot disease caused by Pectobacterium carotoverum. Now, in another set of phages, no, we also looked into 3 phages that were found to infect another species of Pectobacterium, which is Pectobacterium brasiliense. No, now this set of phages were also determined to belong to the family Myoviridae. So because of that typical head and tail structure, we also characterize phages 2, 4, and 6. We determine their burst size, their optimal multiplicity of infection. Now, interestingly, in contrast to the Pectobacterium carotoverum phages, now these PCB phages were observed to have a broad host range. No? So these 3 phages were able to lyse several P. brasiliense isolates, as you can see in this panel. So we have PCB or P. brasiliense isolate 11cc, P. brasiliense isolate 5, 19A, and 19B were all infected, no, or lysed by this phages, no, and then We also conducted the semi-implanta assays, and then what we observed was that these three phages were also very effective, no, in reducing tissue maceration caused by Pectobacterium brasiliense in potato slices. Now, one very interesting results that we want to share is that in our collection, we were able to identify phage isolates, no, having broad host range, no. So we have like six of them, which were isolated using single host baiting, meaning we used a single species of bacterium for phage enrichment. And then three of them were isolated using mixed host baiting. So in this strategy, what we did was to use a combination of bacteria, and then that mixed host was used, no, for enrichment of this phages. Now, interestingly, these phages were found to infect, no, or to show good, very good lytic activity against different species of Pectobacterium in our collection. So in fact, as you would see in this slide, no, so these are the actual results of the spot tests that was done in our lab, and then this would show you the clearing zones produced by this BHR phages in the different species of Pectobacterium that we have in our collection. So very interesting to note was that these phages were found to lyse or infect Pectobacterium odoriferum. And then there were also like 4 isolates of Pectobacterium aroidiarum that was lysed by these phages, and also Pectobacterium cacticida and Pectobacterium carotoverum. No? So let me just mention that broad host range phages would offer good potential for biocontrol application, since as we note, no, there are a lot of Pectobacterium species associated with the soft rot of vegetables. So these would provide us with a promising potential, no, to be used for field application. So at the moment, we still have ongoing semi-implanta assays and screenhouse assays using this broad host range phages that we have identified in our collection. And these are being evaluated or tested against selected Pectobacterium species. No? So we are now focusing our attention, since we have identified the Pectobacterium aroidiarum as the predominant soft rot Pectobacteria associated with soft rot in vegetables. So this would be the focus of our efficacy assays, no, particularly utilizing broad host range bacteriophages. So in summary, the work we did for the work we did in the last 3 years, we were able to isolate 1,268 soft rot associated bacteria from high value crops, no, and only around 22.4% of them were screened as were initially screened as putative SRPs, and then out of the 348 putative SRPs, we found that 163 of them, or that's around 47%, were found to be pathogenic on its isolation host, and we identified Pectobacterium aroidiarum as the predominant species, which constitutes 85% of the total species tested based on 16S rDNA sequencing. Now, it is also worth to mention that we observe significant genetic variation among our identified SRPs based on rep-PCR fingerprinting, as this would be very important towards the identification of phages that would be used for biocontrol against soft rot caused by Pectobacterium. Now, as far as the phage collection is concerned, we have a total of 110 lytic bacteriophages infecting Pectobacterium, and most of them showed high specificity and very good lytic activity, specifically against four species, namely P. carotoverum, P. brasiliense, P. arradiarum, and P. cacticida. And then we also have phages infecting Pectobacterium species with broad lytic activity. So these are our super BHR phages that we were able to isolate. And the semi-implanta assay showed that lytic phages were highly effective against Pectobacterium species, which offers good alternative, no, or as promising biocontrol agents against soft rot. So hopefully, no, in the future, looking ahead at the end of this pandemic, hopefully we would be able to continue with our bioefficacy assays, not only in greenhouse settings, but also in field applications. No, at this point, although we have candidate phages, no, that appear to be very good, no, with very good potential for biocontrol application. Of course, additional studies would be necessary, particularly those that would cover the determination of phage stability in the external environment, as well as the mode of application of phages, no, to control soft rot disease, no, mode of application, timing of application. So these are all still very important things to investigate on, no, looking ahead, no, with as regards the application of phages as biocontrol agents. Now, let me also mention that we would also want to uh, do more studies, no, regarding the phylogenetic analysis of SRPs, as we know that there's still very limited information on SRPs in the Philippines. So hopefully, REC-A gene-based phylogenetic analysis would be very useful for us to understand more about the pathogen associated with the soft rot in vegetables. And of course, no, a study that would focus on the genetic diversity of Pectobacterium using multi-gene sequence typing would also be very essential. Okay, so that's it. Thank you very much for listening. And then let me just acknowledge all the members of our project team, from the project staff to all of the students who were involved in this project. And then, of course, our funding agency, Picardio ST, and the collaborators from the Institute of Wood Science, Entomology, and Plant Pathology, as well as from the National Protection Center of UPLB. That would be all. Thank you very much for listening, and a pleasant morning once again to everyone.
Thank you, Mom Jamie, for that very interesting and very informative talk on the application of bacteriophages on significant crops, on how they can be used in the soft rot of significant crops. So at this point, I will now call on Professor Christian Jordan O. De La Rosa to introduce our next speaker.
Hello. Good morning, everyone. I would like to introduce our second speaker, Dr. Cristel Angelique A. Santiago, was a graduate of BS and MS Microbiology in the University of Santo Tomas. After 2 years of working in the brewing industry, she then transferred to the academe. She was a lecturer for 3 years in Far Eastern University, Manila, and University of Santo Tomas. Fast forward to December 2016, she received a full scholarship from Monash University, Malaysia, where she earned her doctoral degree in applied microbiology in 2020. Her work focused on the biodiversity and metabolomics of lichens and endolichenic fungi. She then planned to continue exploring the metabolomics of fungi because, after all, 3 years of PhD is not enough to learn everything she wanted to learn. She now works in Monash University, Malaysia, as a postdoc research fellow in collaboration with one of the prestigious research companies in Malaysia, the Advanced Agri-Ecological Research. Now, her life revolves around this white rot fungi and metabolomics, and luckily, she's very happy about it. She also blogs at cartoonitall.com, where she tries to combine cartooning and science. Everyone, again, I would like to introduce Dr. Santiago.
Good morning, everyone. I'm Crystal Angelique Santiago, a postdoc research fellow in Monash University, Malaysia. Thank you very much for joining me in this event where I'll be sharing with you a technology which I've been working on since my PhD until now in my postdoc. So today I'll be discussing with you the metabolomics of fungi, a state-of-the-art approach to exploring these wonderful organisms. Now, to begin with, I want to define first what metabolomics is. So actually, I thought of comparing it with solving a crime. So nowadays, we are into forensics, which is a field of science that involves and applies scientific method and techniques to solve a crime or any other mysteries. So this task is not easy to do. It requires expertise, it requires money, equipment, analytical tools, and a systematized protocol to arrive at a conclusion. So unlike before, wherein we hire somebody, we hire a detective to manually track a person by looking for clues, tracing fingerprints or footprints, and all those things. And then in the end, the detective might be able to capture the correct person, or perhaps he may be able to capture the wrong person. So in metabolomics, here I am showing you a comic strips of how to use mass spectrometry. It's actually similar to the mechanism of metabolomics. So for example, you have like hundreds of isolates and you want to identify a novel and at the same time bioactive compounds using metabolomics. So similar to forensics, this one will require expertise, it will require money, it will require equipment, analytical tools, and a systematized protocol to achieve your goal or to arrive at the conclusion. Whereas if we will use the conventional way, let's say again you have hundreds of isolates and you want to find a novel and bioactive compounds. So using the traditional way, you have to test all your samples, do a lot of assays, do a lot of analysis. And then in the end, perhaps you can find your bioactive compounds, but then it cannot guarantee whether the isolates or the compounds that you have are actually novel. So it's like finding a needle in a haystack. So that's how metabolomics works. It actually makes your life in research easier, but when I say easier, it doesn't mean that you don't do anything. It only means that your methods are so robust that your data are very much accurate and reliable. So in a more scientific way, metabolomics is described as a well-established, rapidly developing research field involving global, unbiased, quantitative, and qualitative metabolite assessment within a biological system. It involves interdisciplinary omics approach, which I will be discussing later in this presentation. And it involves analytical platform, multivariate data analysis, and various interpretation of your data. So what's interesting about metabolomics is that it involves computational tools to ensure that all the biases are removed and that your samples are comparable. It also makes use of visualization tools to show trends or patterns in an easy and attractive way. And also there's this one feature wherein, well, they call it the dereplication, which is the rapid identification of known compounds. So say, for example, you have thousands of your samples and you want to know whether it's novel or not. So if you use the replication, it can quickly give you the complete information about your metabolites, and it will also tell you whether it's novel or not. So by doing this, you are actually overcoming the issue on the redundancy and metabolite isolation, which is one of the biggest problems in most researches dealing with drug discovery. So there are actually two types of metabolomics. The first one is targeted metabolomics, and the other is the untargeted metabolomics. Targeted metabolomics is a method wherein you have a specific metabolite in mind. You know what actually you're looking for. It's either a specific metabolite or a specific set of metabolites. Whereas in untargeted metabolomics, it is global in scope. This time you don't have any specific metabolites in mind. What you want actually are all the metabolites present in your sample. So if you search metabolomics in the internet, you could actually see quite a number of publications with this technology, simply because of the following benefits. One is the possibility to rapidly measure thousands of metabolites simultaneously from only a minimum amount of sample. Those researchers who are working in natural products would really appreciate this one. Let's say, for example, you want to have 10 grams of crude extract. We know the pain of like how many samples you have to prepare, how many samples you have to process, and how long the fermentation would take just to get the 10 grams. So if there would be a method wherein you will only use a very small amount of sample and then your data is a lot, then this is something really good. Also, metabolomics offers a comprehensive analysis of metabolites supported by statistics, which in this case is very important for our case because we are publishing papers, and also for those who are into like a patent. And also, metabolomics offers a relatively faster analysis than the conventional method. Now, this is the omics platform, which I mentioned earlier. And this is composed of different omics technologies, wherein first we have the genomics, which is the comprehensive analysis of DNA structure and its function. Proteomics, which is the systematized study of proteins to provide comprehensive analysis of its structure, function, and regulation of the entire system's biology. And of course, we have the metabolomics, which is what we're talking about right now, which provides a functional view of the entire system as a result of the genomics and proteomics. So metabolomics actually is the most recent member of this omics platform. And it was also mentioned in most papers that these omics technologies actually complement each other. So the question now would be, what do these omics technologies tell us? And how do they complement each other? Now, to answer these questions, allow me to show you this illustration. So the gene, the mRNA, the protein, the metabolites represent your genomics, transcriptomics, proteomics, and metabolomics, respectively. So unlike gene, mRNA, and protein, which which are subjected to mutations and different modifications, metabolites, on the other hand, reflects the functional state of the system. So in short, whatever happens to your gene, to your mRNA, and to your protein will be reflected in your metabolites, which is why they say that metabolites actually serve as the downstream products that provide a direct, integrated, and most up-to-date insight into the state of a system, which we call the phenotype. Okay? And from here, we can say that metabolites serve as direct signatures of biochemical activities and are easier to correlate with phenotype or the observable traits of a system or an organism. Now, this is a theoretical metabolic pathway. So what I'm trying to show here is that when you look at the genotypes, an alteration in a single enzyme or a single metabolite in this case can lead to a cascade of metabolic perturbation. So if there is anything that will happen to the genotype, let's say genotype 1, it will be reflected on the metabolites on the lower part, the blue circles. So, and at the same time, from the metabolites, you can actually trace back to the gene, the genotype, to the genes, like what happened, why the metabolites appear that way. Okay, so from here, we can understand that a biologic event should not be viewed as an isolated phenomenon, but as a part of a complex network of changes within an organism. From here, we can observe that metabolomics is actually a significant link to your proteomics and genomics. And then also from here, we would understand why metabolomics actually is a very good field of research, and these can be reflected in various applications where metabolomics was applied. So let's say the clinical research, disease treatment, drug discovery, animal and plant research, agricultural research, and nutrition. So in my presentation, I'll be focusing more on drug discovery. And personally, actually, every time I talk about drug discovery, I always affiliate it with fungi. I think because fungi, I've been working on fungi for quite some time. And I think everybody here knows about, what the fungi are. And currently, there are 5.1 million fungal species known worldwide, but unfortunately, only a small portion of this number have been really explored. And these fungi actually can be categorized into different types. So we have the soil fungi, we have the marine fungi, we have the fleshy fungi, and we have the endophytic or the endogenic fungi. These fungi were explored for their bioactive compounds. Now, I think everybody would agree if I say that the recent fungal researches, which specifically deals with drug discovery, it actually skyrocketed after the discovery of the multi-billion-dollar anti-cancer compound called Taxol. I believe you still remember this. This metabolite was actually isolated from the endophytic fungus Taxomyces andreanae, which inhabits the tree Taxus sprifolia. So the discovery of this isolate actually gave hope to endophytic fungus. It showed that endophytic fungus can actually be an alternative for a sustainable supply of valuable metabolite. And from there on, we then observed a cumulative number of US patents granted on endophytic fungi producing significant bioactive secondary metabolites. And then here, we can actually think that perhaps it's really a good idea to apply metabolomics in fungal research. So in my presentation, I'll be showing you an overview of metabolomics of fungi. Not too much detail, but I just want you to see how metabolomics can actually play a significant role in fungal research. So there are actually 4 steps for conducting fungal metabolomics. The first one is the sample preparation, data acquisition, data analysis, and metabolite identification. So for the sample preparation, you can actually have, you can use different types of samples from blood, tissue, cells, urine, and microbial crude extracts. And you can also use different types of extraction depending on what type of metabolomics you're after, either targeted or untargeted. For the data acquisition, in metabolomics, you may frequently encounter NMR or the mass spectrometry, the MS-based. That's why we have the MS-based metabolomics or the NMR-based metabolomics. So either of these platform can be used for your metabolomics research. But of course, ideally, if you really want to know the global metabolome analysis, then it would be better if a combination of platforms will be used. Now for the data analysis, okay, we have this raw data. You have to remember that these raw data are actually generated from NMR or LCMS. And these raw data, you cannot straight away subject this to data analysis. You have to first do pre-processing and pre-treatment. before you can proceed to data analysis. So the pre-processing, it involves different items. So you have filtering, feature detection, alignment, and normalization. So I won't be discussing too much detail on every item here, but the goal here, the goal of pre-processing is actually to clean your raw data. Okay, what do I mean when I say clean? Okay, I'll show an example. This one is an example of filtering. If you would look at figure A, the, the name is raw spectrum here, you would see that the peaks is actually our LCMS data. The peaks here are very rough, okay? So from here, it's difficult to trust this kind of peaks because you don't know whether the peaks are actually a part of your sample, or perhaps these are just noises. Because you know, in LCMS, there are noises like from the equipment or from the buffer. So you have to do pre-processing to remove all those noises. Okay. So if you would look at B and C, because there are different settings for this one. And also don't worry because this pre-processing can be done using software. Now, in this case, I mentioned here two, the most commonly used, the MZ9 and the XCMS. Now, next would be pre-treatment, which involves centering, scaling, and transformation. Again, I won't be discussing too much on this, but the goal of pre-treatment is to have relevant clean data. Okay, remember in metabolomics, we are actually dealing with thousands, or hundreds or thousands of metabolites. And of course, we want to make your samples comparable. We want it's similar with apple comparing with apple. So we want to do pre-treatment. Next, of course, is the data analysis. So we have the multivariate data analysis. So what's the difference of multivariate analysis with other normal analysis? Is, well, when you read the name itself, it says multivariate, meaning you can have many variables as much as you want. And you can do the analysis in one shot. Okay, so in metabolomics, you will frequently encounter principal component analysis or the projection to latent structure discriminant analysis. There's still other analysis involved. There are also partial least squares, discriminant analysis, and other things. So I just mentioned here the few common ones. And these data analysis actually can be done using different softwares. Here I listed the R project, Metabo Analyst, MATLAB, and Simca. Well, it only depends whether you want the freeware or if you want to purchase the license. Now, also, it's important to note that in data analysis, you have to consider the statistical parameters. Now, in metabolomics, you will be encountering frequently R2 and Q2, which is the goodness of fit and the predictive ability, respectively. So remember that metabolomics actually depends on probability. So everything is statistics. Okay, so these are important. And also we have the p-values wherein it's similar to your ANOVA, t-test, and all those things to validate your data. Okay, these are one of the most important parameters that you have to include every time. Let's say you are publishing a metabolomics paper, you have to include all these things. And lastly would be the metabolite identification and validation. So how do we identify metabolite? So it is actually based on established databases. So here I have listed the most commonly used databases depending on your research. So it could be the Human Metabolome Database, Metlin, or Dictionary of Natural Products. So what's next after identifying the metabolites? This is actually one of the greatest challenge in metabolomics. We should look beyond identifying the compounds. So what's next? We should actually look for biomarkers, correlate these biomarkers to the metabolic pathways, because if you would look at the figure here, this is actually the metabolic pathways, and obviously it's very complicated. So it needs continuous optimization and improvement of metabolomic methodologies. So the question, is metabolomics applicable to fungal research? Of course, the answer would be yes. And in fact, it's not only limited to fungal research. There have been papers about mining of bioactive compounds, chemotaxonomy, plant pathogen interaction, and even human metabolomics. Now, let me share with you, in our group, we actually did LCMS-based metabolomics. This is the biodiscovery of potential biomarkers of an endolygenic fungus using LCMS-based metabolomics. So I won't be sharing the complete details, so I'll be just showing the insights and the outcomes and the results of our study. So we evaluated lichens. In this case, we have the genus Usnea, where I collected, which I collected from Philippines and Malaysia. So lichens, to all those who are not familiar, these are symbiotic association between a fungus and a photosynthetic partner, which could either be a green alga or cyanobacterium. They are known as prolific sources of bioactive compounds, and these actually when you search for lichens, you can find all the biological properties of these lichens. However, despite these benefits, lichens remained neglected by most pharmaceutical companies because of their slow growth in nature. And when we say slow growth, we are actually referring to 2 millimeters in terms of length or diameter per year. So it's literally very, very slow. Also, in our study, we evaluated endolygenic fungi, which are microorganisms living inside healthy lichen thalli. They are analogous to plant endophytic fungi, just that when we say endolygenic fungi, it's inside the lichens and not plants. And they are also known as producers of bioactive compounds. However, this group of organisms are still limited. Now, this map actually proves that. This map shows that there are still very few countries who are really working on these organisms, which actually gives us an edge. Very less competition for our case because it's quite new. And whatever we do, actually, it's one of the few groups perhaps in Asia. Yeah. So the objective of our study is to identify potential antibacterial biomarkers of lichens or endolygenic fungi against Staphylococcus aureus using LCMS-based metabolomics. So again, I'll just quickly share with you our results. So for the first results, we actually tested their antibacterial activities because we will later on use this for the metabolomics analysis. So we found out that lichens and endolygenic fungi are actually effective against Staphylococcus aureus, but only endolygenic fungi are effective against E. coli. Next, we also tried to profile their metabolites. Now, this is an example of a PCA, or the principal component analysis. And here, we observed that the lichens actually clustered together in terms of their metabolic profiles, and as well as the ELF. However, we observed, you see, the XV, which means Silaria venustula, and XSP, which stands for Silaria sp, which are both endolygenic fungi. They are actually separated from the rest of the endolygenic fungi. So then we proceeded to another analysis, which is this one. It's called the loading scatters plot. All the dots that you see here actually represents the metabolites of the samples in the PCA. So here we are only interested in those metabolites that are separated from the rest of the samples. So here, these compounds are actually, we find it very interesting because they are away from the rest of the metabolites. So at this point in time, we don't know anything about the metabolites. What we only know is that what we only know is that Silaria venustula XV is actually an interesting sample. So next, we found potential biomarkers. So how did we do that? This is an example of partial least square discriminant analysis, wherein the dots that you see here are based on their antibacterial activities. So we have the active and we have the inactive. Now in this graph, you would see that the encircled ones, the green, the XV, FR4, these are actually fractions. And from here, we again observe that these fractions are very far from the rest of the samples. So we again have this kind of graph, the loadings plot. And you see these compounds, which are separated from the rest of the samples, are actually very interesting because they are always away from the sample. And then from here, we can say that perhaps these are the compounds which are responsible for their bioactivities. Okay. Now we applied the replication in our study. So in our case, we used the Dictionary of Natural Products, and we tried to identify those interesting compounds that I just showed you earlier. Now, in this stage actually is very critical because from here, this is the decision making of what will we do after. So say, for example, when you look at P60, well, in the replication, you would know all the information about the metabolites. Let's say you look at P60, it is identified as methyl silarate C. And from here, it says that it was first reported in Acilaria. Now, in our study, our isolate is also Acilaria. So we chose not to do anything about it because for our case, we won't anymore do any further studies because there is no more point doing it because it has been reported before. Okay, so instead, what we can do is that we can perhaps focus on, let's say, P844, because here it says that the metabolite was isolated from marine sponge and marine-derived bacteria, whereas in our case, our samples are actually fungi. So these are something interesting. So you see, metabolomics helped us to decide to study further or to stop because we might just be wasting our time and resources. Now these are their chemical structures. Now going back to our objectives, so were we able to identify the potential biomarkers? Of course, the answer is yes. So what else did we find out? Now, in our study, we found out that no further analysis on the lichen host and other ELF are needed. So that's what we found out along the way. We also found out that ELF can be alternative sources of bioactive compounds. And this is the most interesting part because we actually did not really intentionally thought about, discovering the answer to this question. But then, because of our metabolomics data, we realized that there is a distinction of the metabolites produced by the lichen host and endolichenic fungi. This is very interesting because in lichenology and endolichenic fungi, it's actually one of the biggest issues. To the best of our knowledge, we believe that this is the first study which actually has an answer to that issue, whether both the host and the associated fungi actually produce the same or different compounds in a metabolic level. So in our study, actually, we have evidence that they produce distinct metabolites. So from here, we come to believe that ELF, or the endolichenic fungi, contribute to the lichen association by producing different metabolites that may target other competitor microorganisms. So what I'm trying to say here is that metabolomics will not only, because for our case, our goal is only to identify potential biomarkers, but in the end, metabolomics is so, how do you say it? It's very robust that you can actually see other things. You can actually connect the dots using metabolomics, okay? So of course, just like any other researches, the fungal metabolomics has its own limitation, which includes unidentified or uncharacterized compounds. So meaning the databases that we have, of course, it needs to be updated all the time. No single analytical platform can perfectly profile a metabolome because the problem is if you keep using combination platforms, then definitely you will have high cost. And of course, the biochemical pathway and visualization tools are still in infancy. So just a trivia for all of you. So why is metabolomics considered difficult? So again, I'm showing you the omics platform. Okay, so I know all omics technologies have its own difficulty level. So it depends on your perspective. But just by simply looking at the definition, maybe it actually shows why metabolomics is difficult. Let's say, for example, in genomics, we are only dealing with 4 nucleic acids. In proteomics, we are dealing with 22 amino acids. But in metabolomics, we are actually dealing with more than 40,000 metabolites. And I think this one is self-explanatory. The higher the number of entity or elements, the higher the difficulty level would be. Okay. So the take-home message, I hope you learned something from the day. Metabolomics is a state-of-the-art strategy which involves systematic and robust methodologies of exploring fungi, particularly of their secondary metabolites. So instead of just simply looking for the needle in the haystack, maybe if you apply metabolomics, they can actually find the needle in style. So yeah, I hope you learned something from today. Thanks for listening.
All right. Thank you, Dr. Santiago, for that very enlightening talk on metabolomics and its application in studying the wonderful world of fungi, specifically the endolycanic fungi. Now, at this point, we will proceed to our open forum and we will be addressing your questions. And at this point, I will be calling on our moderator for the open forum, Professor Michael Joseph M. Nagpala. Sir Michael?
Sir Mike, nakamute ka. Sir Mike, please unmute.
And sorry, wala po akong ability kanina to unmute. So good, sir. Thank you, Sir Daniel. And good morning, everyone. I will be your moderator for this Q&A session. So we already compiled all your questions from the Zoom chat box and the YouTube comment section. So just in case we run out of time later, we will send your questions to the speakers and we will let you know their responses. So to start the Q&A session, so the first question is for Professor Encabo. And the question is from Ma'am Maria Elareta of Philippine Science Bicol. So for verification, will Pectobacterium species affect production yield of the vegetables that we are commonly consuming? And were you able to report this to the Department of Agriculture for collaboration purpose and action plan?
Okay. Thank you very much. For a moment, I'm just having some issues in my Zoom. Sir Michael, can I request you to repeat the question again, please?
Okay pa, Ma'am Jamie. For verification, will
Yeah.
Pectobacterium species affect the production yield of the vegetables that we are commonly consuming? And were you able to report this to the Department of Agriculture for collaboration purpose and action plan?
Yeah, the answer to that question is definitely yes. No, so just to share with you from our field samplings, no, which we did like from 2 years ago, there were really like a lot of vegetable farms severely affected by soft rot disease, no, particularly cabbage farms and lettuce farms. So in fact, some farmers would really leave their infected crops in the field, and then of course this would result to like so much losses in their part. Now, as far as reporting is concerned, no, so our team is planning to do some sort of communication, no, to concerned agencies as well as local government offices, no, particularly the LGUs that we visited. So just to more or less come up with a very good action plan and also inform them of the results of the survey which we conducted during the time that we did sampling for our soft rat study. So thank you.
So thank you, Ma'am Jamie, for that comprehensive answer. So the next question is for Dr. Crisol Santiago. So were you able to purify and identify the biometabolites? So the question is from Dr. Elzela Bicol of ITDI.
Yeah. No. What I mean is we're currently isolating and purifying the compounds. We're on the way to isolating actually 3 compounds. Yeah. So far, yeah, we're still on that stage.
Okay. So thank you, Dr. Santiago. Next, we have a question for Ma'am Jamie and Cabo. So do pathogens also develop microbial resistance to phage therapy as to chemical management of pathogen?
That's really a very interesting question. No? So when you talk about phage therapy, it's always a story of defense and counter defense. No? So you make use of phages to combat bacteria, but then looking at the way or the rate at which bacteria would evolve resistance, but not only to antimicrobials, but also to phages. No? So this is really a very important challenge. So in fact, there are a number of reports on bacterial resistance to phages because interestingly, bacteria can always become resistant to phages by modifying their cellular structures involved in phage attachment. What else? You also have what you call phenotypic shifts. And then, of course, bacteria can also acquire CRISPR spacers, no? So which are very important defense mechanisms among, particularly for phages. Thank you.
So thank you, Ma'am Jamie. So, just to follow up lang siguro, no? So
Yeah.
will the natural ability of viruses to mutate be a challenge in phage therapy? So that is a question from Ma'am Rina Oplencia of UPLP.
Yeah, so if you look at phage therapy, no, so the dynamics of phage and the bacteria interaction is really a very important consideration. No? So on the side of the bacteria, you have a variety of defense mechanisms that would allow them to become resistant to phages. But then on the phage side, no, probably if we look at phages, no, they have a very small genome size, but despite the small genome size, selection and mutation is still a possibility. No? So there are actually a number of reports on phages, no, being able to expand their genome size and then mutate their receptor binding proteins. No? So if you look at this side of the story, mutation and selection in phages would actually allow them, no, as a way for a counter defense, no, against bacterial resistance. So although these are like recent reports, no, on phage genome size being capable of expanding, but this gives us the idea that that these changes in this bacteriophages would be very critical, no, particularly for therapeutic purposes. If you try to determine or to select which phages are really very applicable for biocontrol applications, no, so you always go back to the importance of understanding phage and bacteria interaction as a defense and counter defense story. Okay, so I hope I answered Mamrina's question. So it's a yes. Thank you.
Okay. So thank you, Ma'am Jamie. So interesting. So parehas silang nagka-counteract with each other. So next we have a question for Dr. Cristel Santiago. So this is a question from Joaquin Valencia of UP Diliman. So what measures have you implemented to deal with the slow growth of lichen? And are there hormones or cell lines currently being developed to speed up the growth rate? So are there other technologies which you believe may help with this issue?
Okay. Thank you for that question. Actually, they are they tried growing the lichen itself, but unfortunately, it is really difficult to grow them. So what scientists actually did, they tried isolating the the microbiome, the fungal partner in the lichen. That's what they tried. But then they found out that the metabolites produced by this microbiome grown in petri dish produce different kinds of metabolites compared to when you grow them, when you just get them from nature. So from here, I think they haven't confirmed a specific technique to grow your own lichen, mostly on microbiome. And how to speed up the rate. Again, for the microbiome part, they added supplements in the culture media to produce more metabolites, but then it's not exactly the same as when it's in nature. For our case, initially I tried working on microbiome, but then it will require a very sterile environment. The technique isn't that super duper difficult, but then it's the probability of you isolating the microbiome, the chances are not that high. So it's kind of risky to do that. Yeah.
Thank you, Dr. Santiago. So the next question is for Jamie. So actually, parehas sa question to from Jess Richardson Gonzalez of YouTube and Sir Ed Leaño. So for commercial application, how will these phages be mass produced and processed for use as biocontrol in agricultural farms? And will the mass production of these phages economically feasible for the field application?
So based on what we did no, in the past 3 years, no, much of the things we focused on was the basic aspect of phage therapy, determining the lytic activities and determining which phages which are more or less suitable for biocontrol applications. So at this point, the proof of concept experiments showed promising results. Now, implementation and field application is another aspect, no? And then usually when you look into the development of biologicals such as phages for field applications, no? So this would still involve a lot of work that would focus on, number one, the mode and timing of application. So before we really go into product formulation and mass production, there are really still a lot of things that needs to be done, no? So our group would really want to move forward, no, towards phages as biocontrol agents. So at this point, I still cannot answer as far as the strategy for mass production and the implementation processes are concerned. But then hopefully, if we move forward with studies that would focus on phage stability, timing, and mode of application, then the information that we would be getting from those studies would really, really be very essential for us to decide on the particular strategy that we would be using for mass production. So I hope I answered that question. Thank you.
So thank you, Ma'am Jamie, for that. So our next question is for Dr. Santiago. So with the countless metabolites produced by fungi in general, how will you zero into the possible novel metabolites or bioactive compounds using metabolomics? And is it enough that a bioactive compound is found in a different organism, or are there any other criteria in choosing which of the metabolites have potential application?
Okay. Okay. In metabolomics, actually, we are only depending on databases. Of course, the statistics that I mentioned earlier, it has to be considered as well. But for the databases, the thing is, there are some researchers who actually stop at showing the bioactivities of extracts. In short, they don't go beyond that. They do not go to the stage of isolating. So in the end, whatever is in the database that's all we have. So there could be a chance that it has been isolated, just so happened, it has been tested, but just so happened that it's not in the database. And another thing is that for our case, we try to look for example, the NMR or the LCMS, and then we try to see as much as possible to look at these databases to see if something match with any in the database. So for now, my answer to that question is that we actually base it on databases per se.
So thank you, Dr. Santiago. So our next question is for Ma'am Jamie, so how will you communicate the technology to the farmer? So how can we convince farmers of the safety of this technology? So the question is from Ma'am Len Balolong of UP Manila.
Okay, so as far as information dissemination is concerned, no, so primarily we would first communicate with our funding agency, no, the important findings that we got from our research on bacteriophages. And then second, since we were able to communicate with local agencies during our sampling, no, so it might be good to go back to the sampling sites that we visited and then coordinate with the municipal agricultural office, which we did, no, during that time of our sampling, and then primarily inform them of the significance of the disease incidence of soft rot in their particular area, and of course, no. The most important thing is to let them understand the phages as an alternative solution to combat bacterial diseases of vegetables, because as we have observed, no, many of the farms that we visited are really using like copper-based chemicals, no, as bactericides and other control methods that are available to them. So the important thing is to let them see the usefulness of phages, no, as an environment-friendly solution to reduce crop losses and hopefully manage soft rot disease of vegetables in the country. So that's it.
So thank you, Ma'am Jamie. And I think we are running out of time, so we will entertain the last question. So this is for Ma'am Cristel Santiago, and the question is from Miss Jessie Gonzaga. So given that metabolomics techniques scans thousands of metabolites, so does it take very long?
I'm sorry, can you repeat the question?
Given the technique scans thousands of metabolites, so does it take very long?
Okay. The reason why we're using metabolomics is because we want to shorten the time. Of course, it will take time, but it would be relatively faster because here, once you have your either LCMS, GCMS, or NMR data, you can immediately screen out. So meaning, depends on what you want. Let's say you have your, for my case, bioactivity. So I can assign them groups in the analysis, and then from there, the software, because basically in metabolomics you're using software here. So the moment that the software will give you an answer to your question. So the important thing that you have to consider in metabolomics, which kind of, for me, it takes so much time, is how to validate your data. So it's like you have to look at the statistics. Of course, it will take time. You have to, because your data, the accuracy of your data, it relies heavily on statistics. And for me, that is quite challenging. It's doable, but it is challenging.
So I think we can entertain another question. So we have a new question here from Zach Lara of UP Manila. So the question is for Dr. Santiago again. So do you think that functional annotation of genomic data from DNA-seq would also provide accurate prediction of metabolites and comparable to the LCMS NMR platforms that you described.
Yes, definitely. For my experience, it's actually better if you have multiple platforms. It will really give you like very reliable data. Yeah, it's possible if the research team has like fun for this one, just go ahead with that thing.
Okay, so thank you, Dr. Santiago, and thank you, Ma'am Jamie and Cabo. So thank you for your questions, and thank you to our experts for sharing their knowledge. So now I will give the screen back to Sir Daniel. So we will forward all the unanswered questions to the speakers.
Okay, thank you, Sir Michael. And again, thank you to our distinguished and esteemed speakers for your very interesting and informative talk. And at this point, for the awarding of the Certificate of Appreciation, I now call on the PSM President, Professor Joel Cornista. Sir Joel.
Thank you very much, Professor Escabo and Dr. Santiago, for that very informative talk. On behalf of PSM, I would like to present the Certificate of Appreciation. May I just request that the certificate be flashed on screen so I'll be able to read it for the, okay. So the certificate reads, the Philippine Society for Microbiology hereby awards a certificate of appreciation to Professor Jamie R. Scabo for having delivered a lecture on bacteriophages as promising biocontrol agents against soft rot Pectobacteriaceae of high-value crops during the PSM Cluster 2 Symposium with the theme Innovations in Microbiology and Biotechnology to Address Global Challenges in the New Normal, given this 20th day of March 2021, signed Dr. Noel Sabino, Cluster 2 Chair, and myself. Thank you very much, Professor Scabo.
Thank you po.
The same certificate is given to Dr. Cristel Angelic A. Santiago for having delivered the lecture on Metabolomics and state-of-the-art approach to explore fungi during this Cluster 2 Symposium, signed Dr. Noel Sabino, PSM Cluster 2 Chair, and myself. Again, thank you very much, Dr. Santiago.
Thank you very much.
All right. At this point, before we proceed to our next speakers, so we will first be showing some videos from our partners. Right. So those are some of the videos from our partners ESCO and 3M. So to introduce our third speaker, I now call on Dr. Marian P. De Leon. Ma'am Marian?
Thank you very much, Sir Daniel. It is an honor to introduce to you our third speaker. Our third speaker is Dr. Eduardo M. Viano. He is currently the Senior Program Officer of the Network of Aquaculture Centers in Asia Pacific, based in Bangkok, Thailand, and manages the Asia Regional Aquatic Animal Health Program. He provides specialist advice on aquatic animal health matters and provides assistance to countries in the Asia Pacific region on the assessment of health status, institutional development, and other activities in strengthening national programs on aquatic animal health management, disease surveillance and reporting, and other health management measures. Dr. Viano serves as Secretary to the Asia Regional Advisory Group on Aquatic Animal Health. He graduated Bachelor of Science in Inland Fisheries from Central Luzon State University, Masters of Science in Fisheries from the University of the Philippines in the Visayas, and PhD in Applied Biology, major in Microbiology from the City University of Hong Kong, Kowloon. He works as a researcher scientist at the Agriculture Department of Southeast Asian Fisheries Development Center, or SIFDEC, in Iloilo, and as postdoctoral research fellow at Taiwan Fisheries Research Institute and National Taiwan Ocean University, Keelung, Taiwan. His fields of specialization are aquatic animal health and aquatic microbiology, specifically marine and freshwater mycology. He has published more than 50 scientific articles and book chapters and co-edited 4 books in aquaculture. He is a peer reviewer of several national and international journals, including Aquaculture, Reviews in Aquaculture, Aquaculture Research, Fish Pathology, Botanica Marina, Fungal Diversity, Indian Journal of Marine Science, and Philippine Science Ethics. Ladies and gentlemen, Dr. Eduardo M. Viano. Sir Ed, the same space is all yours.
Good morning, everyone. First of all, I would like to thank the Philippine Society for Microbiology for the invitation, especially to Dr. Marian De Leon and Dr. Noel Sabino. And it is really an honor to be here with all of you today, even virtually, on this Cluster 2 Symposium of the PSM. So this morning, I will be talking about aquatic animal health management in the Asia Pacific region, focusing on disease surveillance and reporting. The Asia Pacific accounts for about 90% of the global aquaculture production. And the aquaculture industry is considered to be a highly significant food production sector that provides many livelihood opportunities, especially for small-scale farmers, which are common in the region. And it certainly contributes to food security, human nutrition, and poverty alleviation. The aquaculture industry in the Asia-Pacific region has developed rapidly in the last few decades. And although it can be considered that the aquaculture is a fully developed industry, it still faces a lot of challenges and problems, and one of which is disease outbreaks. So disease outbreaks is one of the serious causes of economic loss in many aquaculture operations in the region and around the world. And diseases are mainly a result of transboundary movement or trading of live aquatic animals. And it directly affects the sustainability of the aquaculture industry, especially if these diseases are left uncontrolled or unprevented. Looking at the aquaculture trends in the last few decades, so as we continue to intensify production, uh, more disease outbreaks and disease emergencies will occur. And as we continue to diversify the species that we use for aquaculture, we are increasing the risk of introduction and spread of pathogens, especially transboundary pathogens. And as we continue to diversify the farming systems, we are increasing the potential for spread of pathogens to wider geographical areas and to a wider host range. And the economic and social cost of disease is enormous. In China alone, which is the top aquaculture producer in the world, the estimated losses due to diseases is around a billion US dollars. And worldwide, it is estimated to be around 6 billion US dollars. Over the past 30 years, several transboundary aquatic animal diseases have swept the region of that has caused massive economic and social losses. So if you look at the right side of your screen, I don't know if you can see my arrows. Um, this is some of the aquatic animal diseases that has occurred in the region for the past 3 decades. So the red circles are for crustacean diseases, the blue circles are for finfish diseases, and the green circle, the one at the bottom, is for molluscan diseases. The most recent disease that we have experienced here in the region is TILV, or tilapia lake virus for finfish, and DIV1, or decapod iridescent virus 1 for crustaceans. So the spread of these transboundary diseases clearly demonstrates how vulnerable the aquaculture industry is to disease emergence. And the impacts of these diseases has been aggravated by the lack of effective preparedness and response whenever these emergencies and disease outbreaks occur. And it is now apparent that many of the introductions and movements of aquatic animals have been responsible for the introduction, establishment, and spread of important pathogens or diseases into new geographic areas and host. And once these pathogens or disease became established in the aquatic environment or in the aquaculture facilities and host, such pathogens or disease are almost impossible to eradicate. Therefore, we are encouraging countries, especially major aquaculture producing countries in the region, to undertake aquatic animal disease surveillance and reporting. This is to monitor the important aquatic animal diseases that are present here in the region. So disease surveillance is defined by OIE as the systematic collection, collation, and analysis of information related to the health of aquatic animals. This is in the case of aquaculture. And this is carried out for the detection of clinical disease occurrence or for determination of the presence or absence of pathogens. So I highlighted here the presence or absence because in contrast to disease monitoring, monitoring only involves detection and observation of existing health problems. So if you have a particular disease in the country, then you monitor the spread or the presence of the disease. Whereas for surveillance, even the disease that are not present in the country, you will have to detect the presence or absence of the pathogen that is causing such disease. So disease surveillance or disease monitoring is just a part of disease surveillance. So way back in 1998, this is to encourage the countries to do aquatic animal disease surveillance. The NACA and FAO has launched the Asia Pacific Quarterly Aquatic Animal Disease Reporting System, and the first report was published in the third quarter of that year. So QAAD reporting was established in response to the need of developing a cohesive strategy for aquatic animal health management in the region. It involves collecting and collating of disease data, so the definition of surveillance is here, and it provides insights on the presence or absence of important aquatic disease and animal diseases in the region. So through its more than 20 years of existence in the Asia Pacific region, QAAD reporting has been a useful mechanism for recognizing emerging and important aquatic animal diseases. It provides up-to-date information on important aquatic animal diseases and serves as a guide to participating countries in revising their national list of reportable diseases in aquatic animals. It also serves as early warning system for emerging diseases or pathogens in the region. So if you look at the table here, there are 4 diseases that we have listed in 2005 in QAAD. These are koi herpes virus disease, infection with abalone herpes virus, the white-tail disease. This is caused by the Macrobacterium rosenbergii nodovirus or MRNV, and necrotizing hepatopancreatitis. So after we listed these and encouraged the countries to undertake surveillance to monitor the presence or absence of this disease in their respective countries, these diseases were eventually listed in OIE. In 2007 for KHV and abalone herpes virus, 2008 for MRNV, and 2010 for the necrotizing hepatopancreatitis. And one of the recent diseases that we have listed in QAAD is append. This was listed in 2013. And because of the importance or the big economic impact of append in shrimp aquaculture industry in the region, this has been listed in OIE after 2 years. So QAAD reporting is also a valuable source of information to support risk analysis, so which is needed whenever you introduce aquatic animals that will be used for aquaculture. So risk analysis should be done for such cases. And the presence or absence of the disease, or the information on the presence or absence of the disease that can be obtained from these QAAD reports that we have published, can be used as a reference prior to risk analysis. And QAAD reporting also paved way in the establishment of excellent regional networking in support of disease surveillance and reporting. And through this program, we have established the Asia Regional Advisory Group on Aquatic Animal Health. And this group is still active up to the present time and meet annually to discuss the important issues on aquatic animal health, as well as the revision of disease list for our QAAD reporting system. For surveillance and disease reporting, we used 3 levels of diagnosis for aquatic animal diseases. So I don't want to strain your eyes in looking at the content of this table from Bondadri and Tasso et al. In 2001. So I'll summarize it here. So level 1 includes observation of animal and the environment, and gross clinical examination of the infected or diseased aquatic animals. So this can be done at the farm or on-site or in the laboratory after submission of diseased aquatic animals for diagnosis. And this level 1 diagnosis can be used for presumptive uh, diagnosis of what the disease is. Level 2 includes laboratory procedures, including parasitology, bacteriology, and mycology, as well as histopathology. So this level 2 diagnosis can be used for confirmatory diagnosis of the disease, especially if the disease is caused by infectious microorganisms. Level 3 involves a more sophisticated protocols or procedures and techniques, including virology and cell culture, electron microscopy, molecular biology, and immunology. So this level 3 is also used for confirmatory diagnosis, which is accurate and reliable, but oftentimes it is quite expensive to undertake. So this is the this table shows you the number of countries that used level 3 diagnostics, mostly molecular diagnostics, in disease surveillance and disease reporting in our QAAD reporting system. So I just want to highlight the level 3 here. So at the start, way back in 1998 to 2001, around 5 to 6 countries are using were using level 3 diagnosis out of 15 countries that are submitting the report. And this number doubled in around 2003 and more than doubled in up to 2010. From 2011 to 2015, we're in around 50, still 15 NACA member countries are submitting the report. So 13 out of these 15 countries were using level 3 diagnostics. And from 2016 to the present, when we merged OIE and NACA member countries in the Asia Pacific region for our QAAD reporting, and still we are receiving around 15 reports per quarter, but 93, an increase in percentage of countries using level 3 diagnosis was observed. So long before the COVID-19 or the coronavirus disease, wherein the RT-PCR is considered as a gold standard for diagnosing COVID-19 because of its reliability and accuracy, the aquaculture industry has been using this gold standard for a few decades. This is for the detection of important infectious pathogens of aquatic animals, including viruses, bacteria, and fungi. So we use PCR, conventional, nested, RT, Q, or II. We also use LUMP, or the loop-mediated isothermal amplification, as well as in situ hybridization. So these molecular diagnostic tools paved way in a timely and more accurate disease reporting from most countries in the region. And it provides a more efficient disease, it facilitates a more efficient disease surveillance or pathogen detection, and provision of reliable health certificates of aquatic animal, which is needed for trading or transboundary movement of, especially of live aquatic animals. And also it facilitated efficient monitoring of the spread of emerging and important aquatic animal diseases in the region or in the country. Going back to COVID-19, which we have been practicing preventive measures for more than a year now, including mask testing, wearing of masks, disinfection, social distancing, and quarantine. So we have been doing this also for a few decades for emerging and important diseases of aquatic animals in the aquaculture. So we do testing for important pathogens of fish and shrimps prior to movement or prior to stocking in grow-out culture facilities. And we also do testing for emerging pathogens to monitor the spread of a new disease within the country or the region. We have implemented ban, or for COVID-19, it's a travel ban on transboundary movement of live aquatic animals, especially for diseases that are still non-endemic to the receiving area or country or zone. And of course, we do quarantine for newly introduced aquatic animals prior to use in any aquaculture operations. So let us look closer for two recently recent aquatic animal disease that has occurred in the region and caused significant losses in the aquaculture industry. So these are acute hepatopancreatic necrosis disease, or APEND. We call it APEND for shrimps or cultured shrimps, and tilapia lake virus for, of course, tilapia. So acute hepatopancreatic necrosis disease is caused by bacteria, and tilapia lake virus, of course, by a virus. And these two diseases are present in the Philippines. So the Philippines has been reporting these two diseases from the shrimp aquaculture as well as tilapia aquaculture, since we included these diseases in our quarterly aquatic animal disease reporting system. So for APEND, this is also known as early mortality syndrome when it was first reported in the region, because of the disease caused unusually heavy shrimp or mass mortality approximately within the first 35 to 45 days of culture. Sorry. So the term early mortality refers to the high mortality during the early days of grow-out culture occurring after stocking of the post-larvae of shrimps. So it was first observed in China in 2009, but officially reported in 2010. And Vietnam reported it to be a big problem in the shrimp industry in 2010, also in 2010. It then spread to Malaysia in 2011, to Thailand in 2012. It jumped to Mexico in 2013, and from there it began spreading in other South American countries. And here in the Philippines, it was reported in 2014. The causative agent of the disease was identified in 2013. It is a virulent or a pathogenic strain of Vibrio parahemolyticus containing the toxin plasmid PRA and B. So after the pathogen was identified, molecular diagnostic tools had been developed immediately for the detection of the APEND Vibrio parahemolyticus, which usually targets the PRA and B toxin. However, confirmatory diagnosis of the disease, if there is an outbreak, is still through histopathology that will show you the destruction of the hepatopancreatic tubules, which is caused by the toxin produced by this pathogenic Vibrio parahemolyticus and other Vibrio species which were also found to contain the PRA and B gene, toxin gene. And I want to take your attention to this lump-based diagnostic, which was developed by the team of Professor Mary Beth Bacano-Maningas of University of Santo Tomas. They call it the Jump Alert APEND Detection Kit. So this detection kit is quick, affordable, easy to use, and reliable. But the most important thing is it can be used on site. So you can go to the shrimp farm, bring this minimal materials that you need for DNA extraction and incubation. And in just, I think, an hour, you can get the result whether APEND, Vibrio parahemolyticus, is present or not. So if you want to know more about this Jump Alert APEND detection kit, they also have this for white spot syndrome virus. So you can contact Professor Maningas of UST. And at this point, I would like to thank Professor Maningas for providing me these 2 slides on this jump alert append detection kit. So with experience of the region in several devastating aquatic animal disease outbreaks in the past, so the response to this append by the countries is we can consider it as a bit better than those response that was made for previous outbreaks of emerging diseases such as the EUS and KHV, for example. Because emergency actions by international and regional organizations has been implemented immediately, including regional consultation, emergency regional consultation, and wider dissemination of information through the disease advisory and disease card. And through these actions, many countries or many shrimp producing countries in the regions have been prompted to apply strict biosecurity measures to prevent the entry of append in their shrimp aquaculture industry. So example of which is Indonesia, which is still free from append up to this time. So we immediately listed append in our QAAD reporting system. This is to encourage countries to undertake surveillance and determine the presence or absence of the disease in their shrimp aquaculture industry. Some countries, example Indonesia and India, immediately banned the importation of live shrimps, including PLs or post-larvae and broodstock from countries affected by appends. So here is the travel ban that we are implementing for shrimps because of this append. And the development of molecular diagnostics for append detection has improved the supply of clean broodstock and post-larvae for aquaculture use. And append prevention has been through changes in management, which focus more on environmental or culture environment and management as well as feeding management. Because in aquaculture in general, we discourage the use of antibiotics or antimicrobials. And there has been, especially in the grow out culture of any aquatic animals, and there has been report for this append Vibrio parahemolyticus that it is highly resistant to the common antibiotics that are used for aquaculture. So the use of antimicrobials or antibiotics is not recommended for prevention and treatment of this append. So next we have the tilapia lake virus or TILV. This was first reported in the region by Thailand in early 2017 and by Chinese Taipei or Taiwan in mid-2017. And after we included the disease in our QAAD reporting system, Malaysia, India, and the Philippines reported it in the third quarter of 2017. So in contrast with append, TILV, for TILV, the pathogen is already identified and PCR diagnostic method has been developed and available when the disease is confirmed in the region. Although we encourage countries to report the presence of TILV in their tilapia culture industry, some countries are not submitting any report to OIE or NACA. But local publication reported the presence of TILV in Indonesia, and an international publication just, I think this year or last year, or it is last year, reported the widespread occurrence of the disease in Bangladesh. And again, just like in append, some countries immediately banned the importation of tilapia from countries affected by the disease. So for the regional response, we immediately released a disease advisory once we received the report of the disease from Thailand. And we immediately listed it in our QAAD reporting system to monitor the prevalence of the disease in the Asia Pacific region. An emergency regional consultation was undertaken a few months after the first report of the disease, and intensive training of TILV diagnostics was undertaken more than a year after the first report of the disease. So this was conducted by FAO in collaboration with China. So what's next? So there are still new threats coming. So as what I have mentioned earlier, that as we continue to develop, further develop aquaculture, we intensify culture system, we introduce new species, new and emerging diseases will be coming. And we have to prepare for that. So in our QAAD reporting system, these are some of the recent aquatic animal diseases that we already listed to monitor its presence in the countries in the region. So one is hepatopancreatic mycosporidiosis caused by enterocytozoon hepatopenae or EHP. This is already reported in China, Vietnam, Philippines, India, Malaysia, Indonesia, and Myanmar, and Thailand. And we have viral covert mortality disease of shrimps, which is already present in China, and decapod iridescent virus 1 or DIV1, which is already reported in China and Taiwan. So all of these emerging or problems in the aquaculture industry has already published disease advisory and disease cards. And these are available for free download at our NACA website. It's www.inaca.org. So with this new and emerging diseases and maybe a few more coming in the future, so outbreaks of damaging aquatic animal diseases are likely to continue as we continue to intensify culture production, as we continue to trade and move aquatic animals, especially live aquatic animals, and as we continue to introduce new species for aquaculture. Aquatic animal diseases or pathogen, once introduced into the country or area, is often very, very hard to eradicate. And the risks associated with emerging and transboundary aquatic animal diseases are shared because we have shared water bodies and epidemiological links through trade, especially live movement of aquatic animals. So no country or no province can work alone when dealing with management or when dealing and managing any aquatic animal diseases. And collaborative approach is needed for effective aquatic animal health management, disease surveillance program, and for effective emergency preparedness and response for emerging and important aquatic animal diseases in aquaculture. I think I have to end here. At maraming salamat po sa lahat.
Thank you, Dr. Liano, for your talk with regards to the importance of aquatic animal disease surveillance and reporting. Reminders for our participants, if you have any questions, please do key them in the chat box below. And for now, we'll proceed with our next and final speaker for this symposium. To introduce our final speaker, I will now call on Sir Robbie Vasquez. Sir Robbie.
Thank you so much, Sir Daniel. So it is my honor and privilege to introduce our fourth and last speaker for today. Our fourth speaker is Mr. Angelito Abawag. He is a molecular microbiologist and an environmental scientist. He is currently the Senior Scientist of Microbiome Life Sciences in Singapore, a biotechnology company under KMP Group. So for the past 30 years, he is both a research scientist and a bio-entrepreneur here in the Philippines and in Singapore in the field of microbiome, bioremediation, and wastewater biodegradation. Since 2011, he has been working on portable molecular-based diagnostics in the field of odor and taste detection on water reservoir in Singapore, Ganoderma disease of palm oil in Malaysia, bacterial pathogens of fish in Vietnam, food contamination in food catering and industrial kitchen in Singapore, and viral and bacterial pathogen in Indonesia. He was a final year project external supervisor and mentor for life sciences students from Singapore tertiary academic institutions like the National University of Singapore, Nanyang Technology University, and Nanyang Polytechnic. So Mr. Abawag is a lifetime member of PSM. Everyone, please welcome our last speaker for today, Mr. Angelito Abawag.
Good morning. I am Angelito Ortega Abawag, a lifetime member of the Philippine Society for Microbiology. Currently, I work at Microbiome Life Sciences Private Limited here in Singapore. For this presentation, I will describe the MLS in vitro diagnostic program. I will center on portable PCR, what were the steps we undertook, and the resulting products. I will also talk about non-PCR methods. My goal in this presentation is to make you aware on the various means and application of portable IBD kits. MLS is a biotech company under the KMP Group of Singapore. It is an umbrella group of companies with interest in food production to distribution. We are involved in several industries: animal production, aquaculture, grains, crops, plant oils, ingredients, food processing, distribution, and logistics. MLS my primary mandate is to develop and apply new technologies to enhance productivity among KMP companies. We only started to commercialize products and technologies the last 3 years after testing and applying in our own backyard. Our three main focus as of the moment are: number one, to develop DNA RNA detection kits for animals and workers. Number two, develop portable detection kits for upstream and downstream stages of food production. And three, provide biosolutions for livestock and environmental application. In this presentation, we focus on the MLS in vitro diagnostics. First, let us differentiate for this presentation what do I mean by PCR and non-PCR. Both are methods to amplify DNA. PCR do it through cycles of denaturation, annealing, extension at fluctuating temperatures, while non-PCR amplify DNA without these cycles and uses a single temperature. There are several variants of PCR. Some, like qPCR or quantitative PCR, and RT-PCR, reverse transcriptase PCR, enters the household lexicons due to the pandemic. Other PCR methods are multiplex, nested, touchdown, digital, among a few to mention. On the other hand, non-PCR methods are loop-mediated isothermal amplification, sequence-based amplification, or NASBA, and others. I will not be describing these methods as either many are already familiar with it, or you can go online to read information about it. A short history of the MLS IBD program started when the author led three diverse projects from 2009 to 2015. Primus 25 was a feed additive for pigs. As we are field testing the product, we encounter pig health issues such as diarrhea. In the field, it is very hard to distinguish an early stage effect of enteropathogenic, enterotoxigenic, and enterohemorrhagic E. coli until the symptoms are manifesting. Now, for water reservoir projects, we try to pin down the culprit in making Singapore reservoir water having off flavor and smell. We eventually identified the causal organism, several actinomyces strains, but to check on a daily basis presence or absence of this organism is a very difficult task. Lastly, the oil palm project is about detecting Ganoderma infestation in oil palm. The causal agent is a basidiomycete with fruiting bodies. Unfortunately, when the fruiting body is observable, the infection is on a later stage and intervention too late. Ganoderma must be detected before the emergence of the fruiting body, and given the expanse of the plantation, which is hundreds of thousands of hectares, a portable detection kit method must be used. In all three projects, samples were taken on site, sent over kilometers away to the laboratory, and then do PCR tests. Turnover period from sampling to results ranges from 3 days to 14 days. Now, for research, this is just fine. But for actual monitoring operation, it is far from even be considered. I call it post-mortem results. Imagine, you take a sample of water in a pig farm with suspected enteropathogenic E. coli, and it will take you 3 days to give the result. By that time, the pigs will be terribly sick before even if you identify the causal agent. Now, you take reservoir water in dams to check on actinomycete that causes the off flavor and smell, and you will only know after 3 days whether you have them or not. The water could have smelled badly or had flowed somewhere else. There must be a fast, portable, flexible, and affordable detection kit. What do we need to do? We need to do PCR on site, portable enough to run inside the car. The PCR the PCR run has to be finished in less than half a day. You sample after breakfast, you have the result before lunch. The reagents must be formulated, so all you need to do is add water and the sample. Now, the reagents must tolerate 30 to 35 degrees at least a week. And the platform must be flexible enough to do multiple methods: singlex, duplex, multiplex, LAMP, PCR, RT-PCR, etc. Well, that's tough, but it's doable. Let's move on. We start with the MLS portable PCR-based diagnostics. First, we look for the hardware. We look for several portable PCRs in the market, and we settle in with Mini PCR. It is a product of Ampilus Limited, a Harvard spin-off based in Massachusetts. It started in 2013, and by January 2014, I bought the first unit here in Southeast Asia. Field tested it, corresponds to Sebastian Krebs, the co-founder and co-owner of Ampilus, and some of the feedbacks I gave, they considered to improve the products. We found Mini PCR portable, lightweight, flexible. It can run in AC, in power banks, in batteries. Robust. My first unit is still working. And affordable. An iPhone or a Samsung is more expensive. In 2016, I wrote an article on My Lab in a Box highlighting the use of mini PCR with some projects. In 2017, Sebastian's opening case on his TED Talk is about my experience. These articles and the webs and the TED Talk are all in the mini PCR website. Having a portable PCR is good, but we need to make the PCR reagents portable too. Make sure the target sequence and the primers fits to a T. Make sure the premix reagents are thermostable. Make sure the extraction kit, the amplification premix, and the detection reagents are optimized. Normally, we buy separately the extraction reagents, the master mix, and the detection reagent. All these components must be optimized to ensure good recovery, amplification, and detection. All ingredients of the premix must be in one container: primers, DNTPs, polymerase, buffers, additives. Given all these requirements, MLS came up with a test kit where extraction, amplification, and detection are contained in one kit, fully optimized. It is an ready-to-use format. Once you extract the DNA or RNA, you add it into the dehydrated premix and run. And short and long-term storage in refrigerated condition, not in freezer. And lastly, can tolerate field conditions of 30 to 35 degrees Celsius for at least a week. The last stage of a PCR run is detection. Initially, we solved the issue by getting blue gel, a portable gel electrophoresis with a built-in trans-eliminator, also from the manufacturer of mini PCR. But the troublesome gel cooking, gel setting, adding gel stain, and using ladders that needs freezing storage, plus the difficulty of non-trained or not fully trained personnel in observing gel bands make us shift into a colorimetric dye detection. We use a modified fluorescence method and a DNA enzyme detection using G quadruplex compounds. How do we design a kit? First, we rely heavily on sequencing. Currently, we are using nanopore mean ion sequencer and employ the service of NGS providers like IDT. Second, we do a lot of gene analysis, gene alignments, insertion, deletion, and modification analysis before we even select our target sequence. Third, we synthesize the fragments we target. Currently, we are outsourcing this to a service provider, but soon, with the arrival of our oligosynthesizer, we can do it ourselves. Fourth, we do highly specific primer designs, which we test with our synthetic fragments. And just recently, we are using Cas9 gene editing tools to enhance our productivity. We always have a multi-test standard operating procedure to check our prototypes and design and in doing our quality control. For the case of our ASF kit, we use a colorimetric dye detection. We compare the results with gel band intensity, nanophotometer readings, qPCR values, and finally, sequencing results. What are the features of an MLS kit? We take into example the MLS ASF virus DNA detection kit. It is currently under registration for the Bureau of Animal Industries. The MLS ASF virus DNA detection kit contains DNA extraction reagents, PCR amplification premix, and colorimetric dye reagents. Extracted DNA are added to a dehydrated premix, run in a mini PCR for approximately 70 minutes. After PCR, dye reagents are added to show positive or negative results. In the MLS ASF kit, we design a sequence tail into the primers. If the sample has the target agent, the primers will attach to it. By adding a porphyrin compound and an azynoethylbenzothiazolone dye, a color change happens. The porphyrin binds to the tail of the attached or used primers, oxidizing the dye, which results into a reduction of color, which is otherwise dark green color if the porphyrin is unattached. The amount of target sequence is directly correlated to the reduction of the dye. This is a result comparison of the MLS ASF virus DNA detection kit. We compare color intensity with gel band intensity using gel electrophoresis, A260 and A260 A280 ratio from nanophotometer, and CQ value from a qPCR. Dye color reduction can be observed in the presence of the target sequence of the ASF P72 gene. If there are no ASF virus, the color is dark green, if you can see in the rightmost tube. It means that the dye is not oxidized as the porphyrin do not attach to the primer's tail because it is not used. Now, as you observe the tube from right to left, you will notice the color intensity are reduced. Now, color intensity reduction is due to oxidation of the dye as more porphyrin are attached to the primer tails, which is binded in target DNA sequence. This is an indication of increasing viral load. We verify the result with gel band intensity. There's an inverse correlation between color intensity and gel band intensity. We also compare these results with nanophotometer readings. A260 values are the amount of light absorbed by DNA, while A260 to 80 ratio determines the purity of the sample. A ratio of 1.8 or more is commonly recognized as DNA pure. The higher the ratio value means more DNA, which means more viral load. Using a Biorad CFX96, we determine the CQ value. The value is the number of cycles before we can detect confidently if there are target viral particles in the sample. The lower the CQ value means the higher the viral load. Comparing all results shows the amount of virus present in the sample means reduced color intensity of the dye, increasing gel band intensity, higher absorbance values, and lower CQ values. MLS currently has two kits being introduced in the market. One for the detection of a DNA virus of swine, African swine fever virus, and the other an RNA virus for human, SARS-CoV-2. For both kits, it is being manufactured in a CGMP ISO 13485 certified pharmaceutical company with in vitro diagnostics Class D manufacturing license from the Health Safety Authority of Singapore. We have obtained the necessary licenses and documents for export. These documents are authenticated by Singapore law and the Philippine Embassy here in Singapore. It is now being commercialized in 6 different stages in 6 different countries. We now discuss the MLS portable non-PCR diagnostics. MLS focuses on two different non-PCR detection methods. First, the nucleic acid sequence-based amplification, or NASBA, and the loop-mediated isothermal amplification, or LAMP. In a nutshell, loop-mediated isothermal amplification, or LAMP, is based on autocycling and high DNA strand displacement activity mediated by BST polymerase under isothermal condition. The LAMP reaction is composed of two steps, an initial step of strand displacement and annealing by 6 to 8 primers, and the second step of amplification wherein each displaced strand is used as template to produce more strand, and so on. All these reactions happen in a constant temperature. To compare with PCR, LAMP uses 6 to 8 primers and perform in a single temperature, while PCR only uses 2 primers and run using at least 3 temperature settings. After DNA amplification, the target DNA can be detected by gel electrophoresis or by fluorescent dyes such as cybergreen, ethidium bromide, picogreen, and propidium iodide. Now, MLS has developed a platform of LAMP-based test kit. It is in an 8 PCR tube strip format containing different pathogenic agents such as SARS, non-SARS, human coronavirus, influenza, A and B virus, and other tests. We were able to formulate reagents for simplex, duplex, and multiplex tests. It is of utmost importance the robustness and accuracy of the design primers. Primer design is the Achilles heel of LAMP. Improper primary designs is the main cause of fake positives results due to primer dimers and even octamers. Now, for detection, MLS chooses the appropriate detection method for different pathogens. We employ fluorescent dyes such as cybergreen and picogreen. We also use hydroxynaptol blue and calcine. The MLS LAMP-based platform will be synchronized with gel-based and PCR colorimetric products to provide a wide array of choices for industry clients. One example of industry application would be for viral monitoring for the swine industry. Duplex or multiplex LAMP-based tests will be used for the farms. A PCR-based quantitative colorimetric test would be used for cluster or area testing and for border testing, as well as ports and airports. These are synchronized with qPCR-based validation or confirmation by provincial, regional, and national laboratories. Synchronization is essential so results of different platforms can be validated and standards achieved, which in turn are comparable with international standards. MLS ensures that its designs follow standardized or standard sets by WHO, FAO, and the Office Internationale des Epizooties, the World Organization for Animal Health, or OIE. Another amplification method MLS focuses on is on nucleic acid sequence-based amplification, or NASBA. NASBA is the isothermal nucleic acid amplification of RNA. This is achieved by the simultaneous action of reverse transcriptase, RNA polymerase, and RNase H. RNA are copied when the strand is targeted by the first primer that causes the binding sequence for RNA polymerase, and then use it to initiate reverse transcriptase, which produces an RNA-DNA hybrid molecule. It is then degraded by RNase H. The cDNA binds to the second primer, which initiates the synthesis of complementary strand. The final step is when the RNA polymerase docks to the double-stranded DNA and produces more copies of the RNA. Now, NASBA products are normally detected through gel electrophoresis, but other detection methods are used, such as electrochemiluminescence, horseradish peroxidase, and molecular beacons. Now, electrochemiluminescence, or ECL, uses a probe attached to a magnetic beads to immobilize the amplicons. A voltage increases oxidation reduction reaction that gives a signal and then detected. On the other hand, molecular beacons uses a DNA oligonucleotide labeled with a fluorophore. A hairpin loop forms a quencher which absorbs light from the fluorophore. If a target sequence of the amplicon is detected, the quencher separates from the fluorophore, and as light emission increases, it can be detected by a fluorometer. MLS is developing a NASBA test kit in a microfluidic platform. In a post-COVID-19 world, there would be a need for mobile health testing, especially for international travel and trade. The basic design is to place a NASBA test kit into a microfluidic base to be detected by a portable reader in a dongle, which can be accessed by a smart device. To give you an idea of travel application, we can imagine a traveler purchasing a NASBA kit before departure uh, from the country, from a country. Do the test 30 minutes before arrival and hand over the NASBA kit to an immigration health officer of the destination country. It will be read through a smartphone or a smart device. If pass, the traveler is cleared to enter the country. If fail, the traveler needs to immediately be quarantined and more tests to be done. MLS focus on providing point-of-care detection to industries greatly affected by the pandemic and are vulnerable for future infection. The industries of interest to us are the swine and poultry industry, airlines and shipping, industrial kitchens, markets and trading posts, and occupational health of workers in industrial zones or estates. There are many resources online on the topics I discussed. But for those who are interested to explore the things mentioned in this presentation, the following papers are good to start with. Finally, let me express our heartfelt appreciation for this opportunity, and in behalf of the Microbiome Life Sciences team, have a good and blessed day. Thank you. Good morning. I am Angelito.
Right. Thank you, Sir Angelito Abawag, for your very informative talk on portable PCR and non-PCR based molecular tests. At this point, we'll be having our open forum, and our moderator for this open forum is Professor Cristel Oliveros. Ma'am Cristel?
Thank you, Sir Daniel. So, good morning, everyone. I will be your moderator for today. So, at this point, we have noted and collected all your questions here in Zoom and in YouTube. In the event that we are not able to answer your question live, don't worry because we will forward them to our dear speakers. Let's begin. So, for our first question, this is addressed to Dr. Liano. The question is from Maria Feliciana Eloreta, and I quote, "Can you share with us the latest assessment of aquatic animal disease in the Philippines?"
Yeah, thank you for that question. Aside from the two diseases that I mentioned in my presentation, the Philippines is also reporting for other common diseases for shrimps and finfishes, just like the white spot disease caused by the white spot syndrome virus. This is a big problem in the shrimp industry there. And also the VNN, or the viral nervous necrosis, or we call it the viral encephalopathy and retinopathy. This is a viral disease mainly of marine finfish like groupers or lapu-lapu. So the responsible agency for disease reporting there in the Philippines is BIFAR, in collaboration with their accredited laboratories in other parts of the country like SIFDEC, Aquaculture Department in Iloilo, and the Prawn Producers Cooperative in Bacolod. So I think I cannot recall. I think they also have the infectious hematopoietic and infectious hypodermal and hematopoietic virus, IHHNV, for shrimps. So I think those are the diseases that they had been reporting from the Philippines.
Thank you very much, sir. So for Sir Angelito Abawag, sir, the question is from Stephen Sabinay. He's asking if the mini PCR was bought here in the Philippines. So available ba yan here in the Philippines, sir?
Okay. As of the moment, you have to buy it straight from the US. But our group has just managed to get the exclusive distributorship for the Philippines. We don't know yet what is when would we start distributorship, because it goes hand in hand with our testing kit. the aim of actually of the testing is you don't go to the laboratory as the initial screening. Like what we are doing in our farm, the testing is already in the farm itself, in the farmhouse. Because I think one issue and one learning that we had in this pandemic is that you cannot overburden the technical people. For the case of COVID-19, the health practitioners are the one overburdened because of this. In any epidemiological outbreak, the testing must be done on site so that you reduce the number of samples. For example, you notice that only 3% are positive in any given sampling. So do you mean to say if I have 1,000 sample, I bring it to the laboratory, the people in the laboratory will die? So the initial screening must be done on site. Whatever is positive only goes to the central laboratory. So our main aim also in the Philippines, which we are just starting, is that we bring you have your, I call it the shoebox, PCR in a shoebox. And microbiome's role is to provide the test kit, and we are aligning ourselves with mini PCR. We are also working out a portable reader. I think we will be doing it next year. It's like you have your own qPCR in your car. So the mini PCR is the mini PCR, but the moment you remove your tube, it will be detected like a qPCR. So that the reader, the portable reader, will be coming next year.
Thank you, Sir Angelito. So we're excited for that, sir, no? To finally arrive here in the Philippines, the shoebox PCR.
Actually, there are several units. If you are from Biosci, you talk to Anna Lantican. I gave her one 3 years ago, I think 2 or 3 years ago.
Okay, sir. So thank you. Noted po yan. So for the next question, no, addressed to Dr. Liano. Now, this is from Vinas Gingging. Are there any cases of re-emerging aquatic diseases? Can you give us the factors for its re-emergence?
Yes, there are some cases of emerging diseases. For example, the EUS, or the epizootic ulcerative syndrome. We call it now the infection with the Phanomyces invadans, which is caused by a highly invasive fungus, Phanomyces invadans. It has occurred, a big problem in most countries in the region, including the Philippines, in the early 1990s. And after more than a decade of being a problem, it just disappeared. Maybe a factor that can be attributed to this is the environment, because EUS only occurs during the cold season. So during the cold water temperature, usually from December to February of each year. So I'm not sure if climate change or global warming will play an important factor for that. But most of these pathogens that we are encountering in aquaculture, including the pathogen causing EUS, are opportunistic pathogens. So if you give them an opportunity to attack the host, they will attack. So after more than a decade, this just disappeared. So it doesn't become a problem, but it has spread. It appeared in African region, but in recent years, maybe in around 5 years or so, we had been receiving reports of re-emergence of this disease in some countries in the region, including Malaysia, India, and Australia. So I don't know the reason why. Maybe one possible reason is, of course, the environment. Another possible reason is maybe we have a more pathogenic strain of the pathogen now. That's why they are re-emerging in some countries. But luckily, we still don't have it here in the Philippines or in other countries in the region, our neighboring countries. So this AUS is affecting mostly freshwater fish, especially those in the wild population, including our hito, our dalag, our gurami. So I hope we will not have reemergence of this disease in the Philippines.
Thank you very much, Sir Lianyo. So for another question for Sir Angelito Abawag, this is from Dr. Eduardo Lianyo. Any plans, sir, for aquatic animal pathogens, especially the multiplex lamp, and what is the average cost per test for that test kit?
Okay. Actually, for aquaculture, we are not going for lamp because we found that lamp is good in the laboratories, but the moment you put the lamp into the field, it will go haywire. You know why? Mutation. The Achilles heel of lamp is on primer design. That's why many who have devised lamp for the field application, they have a lot of false positive. The reason for their false positive is actually dimers and octamers, because you are looking at 6 to 8 different types of primers. Now, the moment, and those primers are quite precise. Now, that's also the loophole. If you have a precise primer, and suddenly you have insertion or deletion or modification in your target sequence, your primers will not bind. It will bind by itself. So you produce dimers. Now, if you look at lamp detection is using fluorescent dyes or dyes. Now, the moment you produce dimers, those dyes will bind to your dimers, and you will come up with false positive. Now, what we are doing right now is a modification of NASBA and we are using microfluidics for lamp. Okay, for aquaculture. We also found out that we have within the group of company, we have Pangasius farm in the Mekong Delta. We have prawn farming in Indonesia, and we have finfish also in Indonesia. So you look at it. If you do aquaculture, you take the sample from the water. It is not real time. It is not real time because the water moves. For example, we are doing testing in the Johor Strait, where you have 3 meter per second ocean flow. So how can you possibly say that your sample is real? Right? So you take your sample now, that same water has gone 3 meters away. That is not real timing. So what we are doing right now is like a dipstick. It's like a monitoring sensor they use for chemical water parameters. So basically, the design would be a microfluidic. It will be in place in a tube submerged into the water, and there would be a time wherein the certain part of that water goes to the microfluidic and close it. Then you will have a reading. Then the reading will be monitored by electronic sensor, and it will be sent over by RSS by telephone, whether you have a presence or absence of your target pathogen. We are now field testing it. Aquaculture might quite take some time. Maybe around end of next year, we might have the first prototype for market study. And we are targeting NS. Of course, the perennial white spot, taura. Now for fish, we are more on Edwardsiella, Aeromonas, and the common culprit. And we know that there are several strains right now going around the South China Sea, and we'll try to make our design more robust so it can work out the whole end. I think Dr. Liana knows that different types of certain pathogen, different type. If you're in Thailand, it's different than in Vietnam and in Malaysia. And we are now working on a regional level so that the design will be applicable for at least South China Sea. Yeah.
Yeah,
So the answer is maybe you contact me next year.
yeah. Can I have a follow up because you described the kit to be tested in a moving or flowing water. What about the closed system in aquaculture, usually in ponds or in tanks, which is still the water is moving, but it is within the
Oh yes,
closed
we
system.
have that. Actually, we have a similar system now. We are doing with bangus
Mhm.
somewhere in Iloilo. I will not tell you where. So somewhere in Iloilo, we have a partner there right now. We are developing. The testing now is in the not in the pond itself, but in the gates where the water comes in and goes out. Mhm. So this one, this is not a static testing. It's like it opens up a certain period of time. Then there are micro bulbs, micro gates, so that you do a sampling in a certain period of time. Then you will have your analysis. So it becomes now almost real time, almost real time.
Okay, so thank you for the follow-up question and for the answer, Sir Tolitz. And another question for Dr. Liano from Christopher Aquino of UP Diliman. How do you intend to communicate these important issues on aquaculture to the layman businessman who are not too informed about the science and research in this regard?
Okay, thank you, Christopher, for that question. On our part, as a regional organization, our main contact for our member countries is the government. So for the Philippines, it's BIFAR. So BIFAR is the one responsible to lay down all these informations at the farm level to the layman. And I think the structure of BIFAR now is better in relaying all of these messages in the layman's term because they had all very active stations at all the regions in the Philippines, the 14 regions or 14 plus regions in the Philippines. So they have a local BIFAR station wherein the extension officers are the one communicating with the farmers with regards to disease management, to important diseases that might occur in their culture facilities, and especially in talking to the private sectors of on relaying all the messages that is being relayed from the regional level to the country level and then to the farm level. So in short, it is the responsibility of the government agencies where we are in direct contact with in all of our member countries.
Okay, so thank you, sir. So another question for Sir Angelito Abawag. Sir, in connection lang to the mini PCR kit, no? Is this already being used in the Philippines, this type of technology, sir? And especially in
Uh,
the light of COVID variant testing.
okay. Unfortunately, okay, for our COVID testing kit, we had that last week of January 2020. We start offering it to the Ministry of Health, and we knock on doors on several people, and until now they don't want it. I don't know why they don't want it. So what happened is that we introduce it to companies. Now here in Singapore, the KMP group, we are in the food business, and almost 70% of us of our workforce are Chinese. That's why way back in January 20, 2020, after the Chinese New Year, that year, last year, we are already testing our Chinese nationals. So the intention becomes that instead of selling it to the government or what, we are selling it to companies. So our main clients are actually companies, because if the ruling, especially here in Singapore, if you, let's say I'm meeting with you face to face, I got positive, all the participants of the meeting will be quarantined. And our company controls 60% of all pork product in Singapore. So if our pork unit will shut down, 60% we will not be eating pork because we control 60% of the market. So it is important for our units, working units. So we did not offer, eventually we did not offer it to the government, we offer it to the companies. And there are many companies, unfortunately I cannot tell you why, because of some confidentiality, that are using our unit ever since. Recently, there are army units in the region that are using us because although we are looking at the frontliners, but you see security issue, you're also looking at soldiers. So we are providing them kits, our kit to soldiers. And right now, we are still we are not registering in the Philippines for the COVID. What we are registering is the ASF. So I think we will get our registration within the next few days after the lockdown. Yeah. Because it's one, I think the swine industry in the Philippines is really bad. So we already have, there are already several units in the farms, and they're evaluating it. But yeah, we hope we give them some assistance in this way. We're also opening some of the respiratory panel by May this year. By May this year. Yeah.
So thank you, sir. A lot to look forward from your company, sir. I think the Philippines is in good hands, no, for companies like yours, sir. So again, from a question to Dr. Liano, again from Maria Feliciana Eloreta, how effective are the measures to eradicate aquatic diseases in our country? Do you have any recommendations to BIFAR? Sir, you're on mute po.
Thank you for that question. First of all, as I've mentioned in my talk, once a disease enters the country or area, it's really very hard to eradicate. So rest assured that those diseases that we have there will stay there, and dealing with aquatic animal diseases is quite difficult or quite different from dealing with terrestrial diseases, or livestock diseases, because the aquatic environment per se is a very good medium for these pathogens to thrive. So even if they don't cause the disease, they are there. And as I said, they are opportunistic pathogens, mostly opportunistic pathogens. So once your host is compromised, once you put stress on your fish, on your shrimp, then these diseases will occur. So I think Philippines in general is doing well in terms of management of this disease. So we usually recommend, or the Philippines or the PFAR usually recommend to the farmers to do improved management measures to at least minimize the effect of these aquatic animal diseases. So you can still continue to culture shrimps and fish, even if the pathogens is there, even if some of your fish or shrimps got infected, for as long as you keep the environment, the culture environment, in a good condition, as balanced as possible in terms of microbial population. And sometimes farmers are advised to use interventions such as the use of probiotics, which have been proven to be effective in preventing some of the diseases, or use of immunostimulants that can be added into the feed. This is to increase the immunity or to boost the immune system of your cultured aquatic animals. So diseases will stay there, but we have to live with them, and we have to adjust our management measures, our management strategies in continuous production of our fish and shrimps. So I think that's what the beef fire is also doing, or extending to the farmers there in the Philippines.
Okay, so I think that would be our last question. Am I right? We still have last 2 minutes. Sige. So for the last question, for Sir Angelito Abawag. Sir, from Venice Gingging, what are the common challenges in devising lamp diagnostic kits?
Okay, maybe not only lamp. The moment you devise a kit, you must make sure that it's end-to-end. Okay, whether PCR or lamp. Number one is sample preparation. Okay, in the laboratory and doing research is okay. The whole procedure must be properly optimized. From sample preparation to DNA, RNA extraction to whatever detection you're using. Whether you're lamp, you're using qPCR, you're using normal PCR. The main problem right now in field testing is that those you are buying individual product, and you need to optimize it before you can even do it in the field. In research, that's okay, but the moment you do in the field, you must do that. Number one. So it means if you need to do extraction, you don't just produce lamp. You produce the extraction method optimized for your lamp. That's the first advice. Never separate. It should be together. Okay. Number two, make sure what is your target. If your target is a mutating virus, most likely your lamp will go haywire the moment it goes pink. One example is the African swine fever. Now, the African swine fever, it from one country to another, the virus mutates. Now, consider this: your lamp is very accurate because it has at least one or two sets of extra primer. Okay, that one makes it quite accurate. But what if you have a mutation? What if you have a mutation? One example of a mutation is the ASF variant from Poland and China. There was at least a 300 base pair insertion. Okay, the one being recommended by Spain, the primer by Spain, does not work for China's strain for the simple reason there was an insertion. Now you develop your lamp. You develop your lamp using the old variant. The moment the virus changes, do you think your primers will be accurate? No. Your primers will not bind. The moment your primers will not bind, it will become dimers or octamers at worst. What will happen? You will have a lot of false positive. So you cannot just design it. You must make sure, you must look at the history of the target. If it is a virus, you look at the history as far back as a few generations. Then you do a series of alignments. You have series of genetic studies. Are there insertion? Are there deletion? Are there modification? Then you can only do primer design. Now, in our case, what we do is we go one step forward. We synthesize the fragment itself and see if there are how much modification can it do before we can even do our primer design. Again, the Achilles heel of lamp in the field is primer design. You must make sure your primer design are flexible and robust, or else you're just producing it, but you cannot sell it. The third is detection. Don't limit yourself with fluorescent dyes. Come on. Fluorescent dyes like cybergreen can bind to anything, right? So there are several dyes available in the market already, and there are for experimental uses. For example, in our case, we don't use fluorescent dye for the ASF. We use a G quadruplex compound using a porphyrin and a redox dye. So you have to work on your detection mechanism together with them. And third, make sure your whole premix can sustain field condition. When you say field condition, it's 30 degrees, 30 to 35 degrees, especially if you're doing aquaculture. It's so hot out there. You cannot bring ice pack Your ice pack or even your dry ice will be done within a few hours. So if you're going to do design, you have to consider all these things that I have said. This one is based on experience.
Okay, so thank you very much to our dear speakers for answering all our questions. So we don't, I think, no more time left for the Q&A. So this ends our Q&A session. Back to you, Sir Daniel.
Thank you, Professor Cristel. And again, thank you to our esteemed speakers, Dr. Edward Leaño and Sir Angelito Abawag. At this point, we will be proceeding with awarding of our Certificate of Appreciation to our two speakers. And I now call on the PSM President, Professor Joel Cornista. Sir Joel?
Yeah. Thank you, Dr. Liano and Sir Lito Abawag, whose talks were equally interesting and important. I would then like to present the Certificate of Appreciation, which reads, the Philippine Society for Microbiology hereby awards this Certificate of Appreciation to Dr. Eduardo M. Liano for having delivered a lecture on aquatic animal health management in the Asia-Pacific region, disease surveillance and reporting during the PSM Cluster 2 Symposium with the theme Innovations in Microbiology and Biotechnology to address global challenges in the New World given this 20th of March 2021, signed Dr. Noel Sabino and myself. Thank you very much, Dr. Liano.
Thank you very much.
Okay. Another certificate of appreciation, the same certificate of appreciation is given to Angelito O. Abawag for having delivered his lecture on portable PCR and non-PCR based molecular tests, customizing diagnostics for the industry, given this PSM Cluster 2 Symposium, given this 20th day of March 2021, signed Dr. Sabino and myself. Again, thank you very much, Sir Nito.
Okay. Thank you.
All right. Thank you once again to all our four speakers. We are very fortunate to have been able to hear your talk on your different fields, and we hope to see you again in the next conferences and symposiums that we will be having, or symposia that we will be having. At this point, before we go to our closing ceremonies, we'll first have two video presentations from our partners.
Good morning. I am Paolo Calona, and first I would like to thank Sir Joel and the Philippine Society for Microbiology Board for giving us the opportunity to share with you our brand, Defensyl Isopropyl Alcohol. With health being a major concern of the Filipino consumers, the question that most of us are asking is the question, are all isopropyl alcohol in the market today the same? Because in the advent of a global pandemic, we saw the need for everyday defense. We saw that Filipino consumers use isopropyl alcohol to protect them and their family from disease-causing germs. And this is the reason why when you go to the store, the supply of isopropyl alcohol in the market was limited. And this situation actually forced the FDA, or the Food and Drug Administration, to release FDA Circular 2020-011, allowing licensed drug retail outlets to perform the compounding of alcohol-based hand sanitizers, provided it is done by a duly registered and licensed pharmacist. And this is the reason why we saw a lot of new isopropyl alcohol brands in the market today. But the question is, are you safe with the isopropyl alcohol you are using? And the easiest way for us to distinguish a cosmetic grade versus a pharmaceutical grade isopropyl alcohol is through checking the label. When we look at a cosmetic grade isopropyl alcohol's front label, we can see that the brand name of a cosmetic grade comes first and has the biggest font in the entire label. We can see that the percentage of isopropyl alcohol comes only secondary to the brand name, since this is registered under cosmetics. And just a little note that some isopropyl alcohol are using the percentage not as the concentration of the isopropyl in the product, but as a brand name. But when we look at a pharmaceutical grade's front label, we can see that the percentage of isopropyl alcohol, or commonly called the product's generic name, it is enclosed in the box. It comes first and has the biggest font in the label in a pharmaceutical grade product. When we look at the back label of a cosmetic grade isopropyl alcohol, what we can notice is that in terms of the ingredient, we can see that the full ingredient listing is reflected in the label since the product is compounded to be classified under cosmetics. And just a little note that the order of how the ingredients is presented shows the composition of the product. So if aqua or water comes first in the ingredient listing, then the composition of the product is mostly water and not alcohol. And the most important thing that you need to look at is the DRHR number. DRHR, or Drug Home Remedy Number, is issued by FDA. It is nowhere to be found in a cosmetic grade product. This is only seen in a pharmaceutical grade product. Looking at a pharmaceutical grade isopropyl alcohol's back label, we can see that a pharmaceutical grade product contains pure isopropyl alcohol concentration, thus reflecting the amount contained in the bottle. And again, the most important thing that will separate a pharmaceutical grade from a cosmetic grade product is the DRHR number. DRHR number is the FDA product registration number, showing that it passed the strictest FDA standards. So the next time you choose your isopropyl alcohol, don't forget to check the label. Again, thank you from Defensal Isopropyl Alcohol, a brand recommended by the Philippine Society for Microbiology because it kills germs on contact, kills 99.9% of common germs and bacteria, and was tested to give you protection for up to 12 hours.
Ang mga panlabas na damit ay pwede magpasok ng dumi at germs tulad ng virus. Paalala ng DOH, sama-sama, let us all be the solution sa COVID-19. Balik trabaho na ang maraming Pilipino, pero ayon sa Philippine Society for Microbiology, ang germs tulad ng virus ay pwedeng maging pasalubong natin sa bahay.
Ang germs tulad ng virus ay maaaring kumapit sa ating damit panglabas at maaari nating mauwi sa bahay ang mga mikrobyong ito. Kaya payo ng Department of Health, ugaliin mo ba rin agad ang panglabas na damit at ihiwalay sa ibang lalagyan para malabhan sa lalong madaling panahon. Mukha mang malinis ang damit, maaaring mayroon itong viruses at posibleng may invisible stains that can feed germs. Isa sa mga paraan para matanggal ang mga germs tulad ng viruses na ito ay gumamit ng detergent na may bleach na kayang sirain ang outer layer ng germs tulad ng viruses.
Laging tandaan, sundin ang payo ng DOH at ng mga eksperto na ihiwalay ang damit panlabas at gumamit ng detergent na may bleach. Be the solution sa COVID-19!
Right, so those are the videos from our partners, Defensil and Ariel. At this point, we will be proceeding with our closing ceremonies. And to start off, we will first have the oath taking of the new PSM members. Now, reminders for the new PSM members, kindly open your cameras and you will be facilitated by our PSM President, Professor Joel Cornista.
Okay. Good morning, everyone. Can you please raise your right hand and repeat after me? I state your name.
I, Christian C. Ramos.
Having been accepted as regular member.
Having been accepted as a regular type member.
Of the Philippine Society for Microbiology Incorporated.
Of the Philippine
Society for Microbiology
Incorporated.
Do hereby swear.
Do hereby swear.
Do hereby swear.
That I will uphold my profession as a microbiologist.
That I will uphold my profession as a microbiologist.
And faithfully discharge my duties and obligations.
and faithfully discharge my duties and obligations
to the best of my ability.
to the best of my abilities.
That I will support the activities for the upliftment of the profession
That I will support the activities for the upliftment of the profession
and for the protection of the rights and privileges of a PSM member.
for the protection of the rights and privileges of PSM member.
That I will do my duty towards God,
That I will do my duty towards God,
my country, and my fellow men.
my country, and my fellow men.
And lastly,
And lastly,
that I impose upon myself the aforementioned obligations
that I impose upon myself the aforementioned obligations
voluntarily and without mental reservation
voluntarily and without mental reservations
or purpose of evasion.
or purpose of invasion.
So help me God.
So help me God.
Okay, congratulations everyone and welcome to the PSM family. Please don't forget to sign your oath of membership and forward it to the Secretariat. Again, thank you very much for making PSM a society in which you trust and become a member of. Thank you very much and congratulations.
All right. Again, congratulations to the new PSM members. And now we will proceed with the awarding of the Thesis Reproduction Grant. And reminders again for the awardees, kindly turn on your cameras. And this Thesis Reproduction Grant will be led by or will be awarded by Professor Angela Melissa A. Carlos. Amilet?
Hi, Sir Daniel. Thank you so much. Good morning, everyone. I hope you can hear me. On behalf of the Philippine Society for Microbiology and the TRG team composed of yours truly, Dr. Leslie Dalmasio and PSM President Professor Joel Cornista, I present the Thesis Reproduction Grant to Clariel Dane Q. Llenado and Zachary B. Lara from UP Manila. The pair is currently working on their thesis entitled Profiling the Rhizostome of Summers Unexplored Karst Forest Soils using Shotgun Metagenomic Sequencing under the mentorship of Dr. Marilyn P. Balolong. Guys, are you around? Can they be spotlighted? So I can see Clariel. Clariel was your partner. Zachary. Okay, so congratulations, Zachary and Clariel. Now, let me just announce that slots are still open for the PSM TSG and TRG, as well as ESCO's thesis support grants. Please check the PSM website for more details and submit soft copies of your documents to my email address, that is aacarlos@up.edu.ph. Thank you. And now, back to our host.
Thank you, Ma'am Millet, and thank you once again or rather, congratulations to our TRG awardees. Right? So at this point, we'd like to thank the Institute of Biological Sciences. It wouldn't be possible, this webinar wouldn't be made possible if not for the support of the Institute of Biological Sciences, UPLB. Also, we would like to thank our partners, PNG, ESCO, 3M, Defensil, and Ariel. Also, we would like to invite you to the 8th Asia Pacific and Biotechnology Congress and the 50th PSM Annual Convention and Scientific Meeting with the topic, Microbiology and Biotechnology, Scientific Opportunities to Address Global Challenges. And for more information, kindly visit the PSM website. For our closing remarks, I now call on the chairperson of this Cluster 2 Symposium, Dr. Noel G. Sabino. Sir Noel?
Thank you very much, Sir Daniel. Okay. Before we formally close today's event, I would like to take this opportunity to express my sincere gratitude to our distinguished speakers, Professor Encabo, Dr. Cristel Santiago, Dr. Eduardo de Año, and Sir Lito Abawag, for gracing this occasion despite their very busy schedules and in sharing with us through their excellent presentations the innovative technologies in the field of microbiology and biotechnology. I would also like to thank the Institute of Biological Sciences for serving as our partner in this endeavor. To the members of Cluster 2 Committee, Dr. Lucille Villegas, Professor Christian Jordan de la Rosa, and Sir Robbie Vasquez, and to the board members of PSM, thank you very much for all the help extended in planning and holding this online symposium. Acknowledgement is also due to our sponsors, Procter & Gamble Philippines, ESCO Philippines, 3M Philippines, and Defense Seal for the support to make this symposium possible. Finally, thank you to all the participants for taking the time to attend the symposium. I hope that by listening to the discussion and exchange of ideas, you have gained a deeper understanding of the important role that scientists, specifically the microbiologists, play in making people's life better under the present situation, which we refer to as the new normal. Mabuhay and keep safe always. Sir Daniel?
Thank you, Sir Noel. And again, thank you to all the participants who attended this Cluster 2 Symposium. And for our certificates, again, you are to accomplish the evaluation form that we will be showing in the screen. There you go. So to get your certificate, please answer the survey below, and it will be made available also, I think, in the chat box. All right? So again, to get your certificate, please answer the survey below. www.psm.org.ph/PSM webinar day 1 eval. All right. So before you close the Zoom, we'll have a photo opportunity. So I'm kindly requesting everyone to please turn on their cameras so that we'll be able to take a photo of this event.
Sir, hindi po ma-turn on lahat ng cameras.
Hi, Sir Robbie, would you be able to turn on or allow them to turn on their cameras, perhaps?
Yung akin din po.
Let's just wait. We'll just have some. We just have some technical difficulties. So let's just wait for a bit. Hintay lang po.
Sir Jed, yun din pong link na binigay. Pupunta lang po sa website ng PSM. Andun po ba, sir? For evaluation.
Maybe kakaproblema
okay na po
tayo.
ba yung mga video? Okay na po ba yung mga videos?
Ayaw.
Ayan, nag-turn on na po yung mga videos. Ayan, nakikita na po natin.
Yan.
Okay na po. I think okay na po lahat.
Ayan.
So Hello everyone. I will be the one to take the screenshot. So when I say smile, can you please hold your smile for 10 seconds because according to the gallery view, there are 12 screens because there are 283 participants. Okay? So 10 seconds. Please hold your smile for 10 seconds. Okay. Are we all ready? Okay. One, two, three. Smile. Next. Okay. Thank you, everyone.
All right.
Thank you,
Thank you, Sir Christian.
everyone.
And again, thank you to all the participants. And for the link po, wait lang po ka sa link, ilalagay po sa chat box. All right. Again, thank you to our speakers and to all the participants. And we hope to see you again in the next symposium.
Thank you, everyone. Congrats, Cluster 2.
Thank you, everyone.
Thank you po.
Thank you po.
So much more.
Ang mga panlabas na damit ay pwedeng magpasok ng dumi at germs tulad ng virus. Paalala ng DOH, sama-sama, let us all be the solution sa COVID-19. Balik trabaho na ang maraming Pilipino. Pero ayon sa Philippine Society for Microbiology, ang germs tulad ng virus ay pwedeng maging pasalubong natin sa bahay.
Ang germs tulad ng virus ay maaaring kumapit sa ating damit panglabas at maaari nating mauwi sa bahay ang mga mikrobyong ito. Kaya payo ng Department of Health, ugaliin mo ba rin agad ang panglabas na damit at ihiwalay sa ibang lalagyan para malabhan sa lalong madaling panahon. Mukha mang malinis ang damit, maaaring mayroon itong viruses at posibleng may invisible stains that can feed germs. Isa sa mga paraan para matanggal ang mga germs tulad ng viruses na ito ay gumamit ng detergent na may bleach na kayang sirain ang outer layer ng germs tulad ng viruses.
Laging tandaan, sundin ang payo ng DOH at ng mga eksperto na ihiwalay ang damit panlabas at gumamit ng detergent na may bleach. Be the solusyon sa COVID-19!
Ayan, tayo na lang.
Sir Noel.
Congrats, Cluster 2, Sir Noel, Ma'am Lucia.
Congrats, Sir Noel.
Lorian, ang Robbie.
Tama po sa tulong.
Congratulations!
Salamat po sa tulong.
Congratulations, pasito raw kayo.
Magpasito daw kayo.
Sir Noel, pamerienda naman yung mga moderators natin, sina Sir Daniel, Mike, at saka Christine.
Ay, ako na pong bahala doon.
I-reimburse na lang
Ako
ni
na pong
Tita
bahala doon.
Orcel.
Opo, ako na bahala doon.
Kawawa naman, gutom na rin sila. Tita
Oo
sabihin
nga,
mo na
dapat
Tita
meron
Orcel,
din
isama
sila.
doon sa reimbursement ng Cluster 2.
Okay.
Sir Noel, pakicompute po ng mga ipapadala namin sa iyo kasi magpapaload po si Reggie ng Gcash.
Ano po? Ay, hindi ko po marinig, Ma'am Orcel.
Pakicompute po ng mga ipapadala namin sa iyo for honorarium lahat. Yung sa honorarium.
Lahat, pati merienda.
Okay pa.
Naman po siya